Okay, we are live. Hello, everybody. We have just touched down on an unnamed outcrop east of Caballito. We're calling it Unnamed Outcrop east of Caballito. We are the first people to see this outcrop, so we're happy to have you with us. Today is June 11th, which means it's a special day for us here. Not one, but two of our pilots are celebrating a birthday today. We have Carl Hale, Chill, and Jason Rodriguez, J-Rod. Today is their birthday. So please join me in wishing them a happy birthday for this dive. Also in the control room, I have with me Emily Cowell from Temple. She's our watch lead, and she will introduce you what we're doing here and what our dive goals for the day are. So, Emily, take it away.
Hi, everyone. I'm Emily Cowell from Temple University. I've also got with me Holly Lutz from Scripps and Rachel Lauer and Rob Perrin from the University of Calgary. Our plan today, this is a newly mapped out outcrop. We've been doing some seafloor mapping of the last couple of days, and we've picked some waypoints to go to. This is an outcrop that's had some previous heat core measurements done from heat flow measurements done from a ship, where they'd sort of almost like a gravity core, they drop the probe down from the surface. So the focus of today is going to be doing some more heat flow measurements to see if this does appear to be an area of hydrothermal discharge. That's what was indicated by the previous heat flow measurements. So we'll be going along to various waypoints, doing heat flow in these sedimented areas, and then also just keeping our eyes open for any signs of venting, collecting temperature measurements, having a look for target organisms as well. So, is the first thing we're doing here at this waypoint just a heat flow?
Yep. Cool. So for the first one, there's going to be an extra 240 seconds before insertion. So we'll just tell you when to insert.
Okay. Sounds good. So we'll just do it right around here, I guess. I mean, are we actually at the? Yeah, we're at the waypoint. So
They'll check to make sure we can get the penetration.
Absolutely, Dr. Meyer.
Really for your ship.
It's a little bit more.
Make sure you ask me for what you want me to label it as, and then I should be able to. Yeah.
Yeah, so our plan is, since we're in a sedimented area, that means we can actually insert the heat flow probe correctly, hopefully. Hopefully the sediment's deep enough here. It looks like it probably will be. So they're just extracting the probe now. We're calling it Excalibur.
Angela Hoover.
Yeah.
That one.
And it's the midpoint of the cruise.
Yeah.
Very interesting day. It's just everything's happening at once.
Sorry, not saying anything. I'm responsible for all
jinxing.
So yeah, we're going to just touch down on the sediment here, and we'll first insert the probe to check that it actually goes all the way, the sediment's deep enough that it goes all the way up to the top marker. And then after that, we'll do the heat probe measurement. So we'll be here a little while doing that.
Going out. I've seen them doing that.
Oh, I've seen that. I'll find it.
It's quite hard, so.
Interesting. Two stuff.
You fooling me around, or is it?
I'm trying.
Because I'm not. All I'm doing is thrusting down.
Does it need to cover that top marker as well?
Go back up.
Ideally, yeah.
Should try a couple
It's
of
pretty
different
hard.
Take it
places.
down. I think
or-
you're just pushing me around.
Yeah, please put, tighten it hard.
Yeah.
Yeah.
Oh, interesting.
Mm.
Yeah,
It's,
Oh.
that's why we're fighting.
That looks a little bit tight.
Is it bent?
A little bit.
There's a little bit back there.
Yeah.
That's the best.
So.
And I'm gonna try another place just in case it's, sure.
Yeah, I mean, I think the way the probe goes in is so perfectly straight. Maybe it, I don't know. That's scary. But
Mm-hmm.
what's Excalibur made out of?
Stainless, I believe.
Then against it there, that's pushing the whole lot of the off.
Wow.
Okay.
It's a really compact sediment.
So it's like some kind of hard layer under.
Well, you see the white? I think
Yeah,
that's what.
we're hitting.
Yeah, that we've collected some of that before. It's real. I mean, it's not rock, but it's pretty dense.
Yeah. It's like choppy. Yeah.
It's fairly pushing a lot of the off there.
And it's hitting at sort of the same spot. It's like,
It is, yeah.
That's like, well, I don't know what it is, it's gonna be 10 centimeters,
yeah,
20 centimeters down.
yeah.
10, 20.
Yeah.
Yeah.
There it is.
Yeah.
20 centimeters and it's, looks nice. Go to the next one.
Yeah, I mean, let's head to the next waypoint.
Okay.
And I mean, in the waypoints are a guide. If there are places along the way there are more sediment and a better place to do a measurement, that's fine.
Okay.
'Cause we-
Just start flying the line then, and we'll try again.
Yeah,
Maybe halfway between the waypoints. We
Yeah.
could try again. Something
or
like
just
that.
or just wherever we see something that looks different than this.
Right.
This,
Okay. Okay, yeah, so it's looking at about 20 centimeters under the sediment. There's a very dense white layer, which is might end up damaging the probe, I think, if we force it. So we're going to just continue from here,
Should get a decent.
head off in the direction of the second waypoint, and see if we see any sediment that looks a little bit different. Maybe test a couple of spots as we're going along.
So, locations. So we go past this one. We might be able to get higher towards the outcrop, so it might even be better.
we're gonna be. It's gonna be a struggle to find peak flow.
Right.
For us, it would have been better to be on the
Very
stuff,
sedimented. Yeah.
but it's boring for biology, so we're kind of just.
Want to look at that?
I don't want
second
to
camera?
start, but yeah, can we just take a look? Because this is.
Can can we just take a little zoom in on the sea cucumber as we're heading across? Thanks.
So we're heading down into this. Hopefully this is better.
This is.
Yeah, can we go like that? Is that way? Is that?
I think there's a limit of a couple of sea cucumbers a die, so I feel like
we only
it's
got one
up to...
yesterday. Would you guys mind if we collect this?
Yeah.
You can go into Coral Quiver. This is great.
You can do that, or
Yeah.
what I want to see you guys do?
That's a species we haven't gotten that one yet.
Just barely grip it.
With a
I know. I just, well, no one did anything yesterday.
needs to...
Green.
I think there's a... I'm sure she said there was a guy, a couple of sea cucumbers died.
Yeah.
So, at the end of the day, that's the one you want.
yeah.
You guys will
See how this reacts.
It has colors like human.
Yeah, it does. Are we putting the heat probe away? Is that what we're up to right now?
We're gonna use the other arm.
Oh, okay.
We gotta get a Coral Quiver open first.
Okay. Just we would need, I think, a zoom and a lasers on and off kind of thing before we collect. Thank you. Yeah, there's some interest on this cruise in collecting sea cucumbers and assessing their microbiome. So we're aiming for a variety of different species and representative of sea cucumbers from across the different sites. I'm being told this one looks like one that we haven't collected yet, so while we're here, we might as well make a quick collection of this, and then we'll be on our way trying to find a spot with maybe thicker sediment to do the heat probe measurement. So before we do any collections, we always like to get some nice images of the organism because they never look quite the same once they're up on deck. Little crustacean on it as well. There we go. So we like to do one sort of glamour shot with nothing else in frame. And then we also like to do one with the scale lasers on. And that just gives us a good idea of the size of the organism.
Sorry, just playing with the camera.
Sure, no worries.
Hurry before it swims away.
Yeah, somehow I think we're okay. Slowest speed chase ever.
It's just a crustacean of some kind. I don't know, it's a little shrimp.
Yeah, looks like it.
I don't
I
think
don't
so.
know. Looks like it's just chilling on it.
Yeah, I haven't seen them on other companies. It's probably just happens to be sitting on them.
Yeah, maybe.
But I learned yesterday that there's a family of fish that cucumbers will often have like little fish that live inside them.
Some layers on it.
Yeah.
Oh, I knew that about in shallower waters that they like
No,
live inside
Yeah.
the sea cucumber.
do you remember the name of these? No. Okay.
So you can just see in the view the two green lasers there. Those are 10 centimeters apart, so they give us a good reference for size of the sea cucumber. Looks like it's probably a 15 to maybe 18 centimeter long sea cucumber, something like that.
Good.
Great. So this will be going into coral cover 1. I've been really impressed by the colors of these sea cucumbers so far.
Yeah. Yeah. Me too.
I feel like my reference point of sea cucumbers is normally that they look quite boring.
Yeah.
But they've been amazing. The little hitchhiker doesn't want to leave.
Yeah, it'd be kind of nice if it stays there.
Yeah.
2 for 1.
Wow, the colors underneath are amazing. So sea cucumbers are typically deposit feeders. So they go along the sediment and actually eat the sediment, the sort of surface layers of the sediment. And so often you'll see these little sediment kind of coils across the sediment, across the surface of the seafloor. And that's what comes out the other end of the sea cucumber after it's finished extracting any good nutrients it finds. Oh, it's trying to make a break for it. Ooh. Ooh. Oh, I think it's got itself turned around. Oh wow. Oh. 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 Goodness me. See, maybe you were right. Maybe this is going to be quite a dramatic chase. Let's see. got pretty good escape plan, this guy.
Yeah, yeah,
I mean, he did well, to be fair.
alright.
Looks like he's in there now, I think.
Yeah, I think so.
Yeah. Give him credit, he was putting up a good attempt there. Good save. Well done. Okay. So in that case, on that note, I think we'll keep transiting towards the second waypoint. We'll see if we can find any deeper sedimented areas for the heat probe.
So we can see this white material kind of scattered in here.
Mm-hmm.
I guess that would be a good telltale sign. If we stop seeing that, maybe it'd be a good time to check.
Yeah. Yeah, for the question in the chat, the containers on the ROV are not pressurized. Actually, organisms such as sea cucumbers, they typically aren't alive by the time they come up to the surface for most of the organisms coming up from these depths, but it's actually the temperature change that tends to have the biggest effect in that. Organisms like these, they don't have any sort of air pockets in them at all. So the pressure often sort of doesn't have the dramatic effect that you would expect. Those are look like rocks. Ooh. Just a fun little swimming polychaete there by the looks of things. We've seen quite a few of those on the descent. Lots of brittle stars again on this dive. You can see the sort of difference between a brittle star and a normal sea star. Brittle stars are ophiuroids. Sea stars are asteroids. But you can actually see brittle stars kind of pulling themselves across the sediment with their long arms. They have this larger central disc and then typically kind of skinny arms. Whereas the sea stars move around using their tube feet systems. So they seem to sort of glide around a little bit more. So I can't remember the name of these.
But I don't remember the sign.
Mm-hmm. Yes, we've seen a lot of these sea cucumbers on previous dives. I think we've collected one of these guys. The common name is the sea hare. Yeah, very cool. These really interesting projection at the back.
This one.
So you can see just there what I was mentioning about the brittle stars kind of pulling themselves along on the sediment with their arms. We tried to do we're between waypoint 1 and 2. We tried to do a heat flow, but there's about 20 centimeters down, there's a kind of white layer that's too hard. It kind of bent the probe a little bit. So we're looking for
the next waypoint.
yeah, and I think in between times, maybe stopping a couple of times, seeing what these guys think, but seeing if we can find another spot to do the heat probe. Cool. You ready to jump in? Okay. So that's the end of my dive shift, but I'll be passing over to Beatrice from the University of Costa Rica, so she'll introduce herself. Good morning, guys.
Good morning, everyone. My name is Beatriz Naranjo. I'm from the University of Costa Rica. I will be joining you for the next hour. I'm gonna switch to Spanish because I want to speak with my Costa Rican, Latin American folks. So I will switch in between Spanish and English. Buenos días, mi nombre es Beatriz Naranjo de la Universidad de Costa Rica. Voy a estar acompañándolos en la siguiente hora. Ahorita estamos explorando un sitio que aún no tiene nombre. Hemos estado viendo algunos peces como ese que teníamos en pantalla, que es de la familia Ophididae. Algunos costarricenses latinoamericanos lo pueden conocer como congrio rosado, o bueno, la misma familia que el congrio rosado, una especie de mucha importancia comercial. Esta es otra especie que vive más profundo. Es una familia de peces que es bastante importante en los fondos profundos. Son de los principales depredadores que vamos a encontrar en estas aguas. Acá tenemos otro en pantalla. Aún no estamos seguros de la especie, tenemos que revisar bien los videos, las fotos. Son grupos difíciles porque aún conocemos muy poco de nuestra biodiversidad de aguas profundas. Poco a poco hemos ido recopilando investigaciones que nos permiten poder ir identificando todas estas especies. Hemos visto muchos pepinos de mar, como ya les hemos informado a lo largo de la expedición. Estos son indicadores de estos sedimentos con gran cantidad de nutrientes. Y ahorita lo que estamos haciendo es principalmente haciendo mediciones de flujo de calor en estos sedimentos. Tenemos un equipo acá a bordo que incluye una geofísica y un hidrogeólogo que están estudiando todos estos flujos de calor. Para explicarles un poco de qué se trata, en los montes submarinos y en otras exposiciones de roca se crean puntos de acceso donde el agua de mar puede penetrar hacia la corteza terrestre, y en esos puntos donde ingresa el agua, esta agua se empieza a calentar por el mismo calor de la tierra y puede atravesar varios kilómetros y descargarse en otros sitios, como usualmente otro monte submarino o inclusive el mismo monte submarino, y se libera al ambiente nuevamente, viniendo de la corteza con unos grados, o sea, un poco más caliente de lo que había entrado. Entonces, ahorita cuando paremos a hacer pruebas, Vamos a intentar ver la profundidad del sedimento para asegurarnos que el sensor que vamos a poner en el sedimento pueda accesar sin problema. Y dependiendo de esa temperatura que nos indique, aquí los investigadores a bordo, especialistas en este tema, pueden tener evidencia de si este es un sitio de recarga acuífera hacia la corteza o más bien un sitio de descarga. Si la temperatura del sedimento es más fría de lo esperado, es un indicador de que el agua está entrando, y si está más caliente es un indicador de que el agua viene saliendo. Entonces, parte de las investigaciones que estamos haciendo, porque se cree que este flujo de agua y esos cambios de temperatura son bastante importantes para los sitios donde hemos estado observando pulpos incubando sus huevos.
Yeah, it's, we bent the ice now, we're trying to put it already. This hopefully in a little bit.
Okay.
Yeah, thank you.
Okay. Beautiful. Este organismo es emparentado con los corales, aunque no parezca. Hemos estado viendo algunos bastante bonitos. Este organismo creemos que es de la familia Corimorphidae. Es una familia de hidroides. So what we're seeing is a Corimorphid hydroid. It's a Nidarian in the same group of the corals. They resemble like small flowers, right? Really beautiful. Can we put the lasers for a bit?
Yeah.
Thank you.
Maybe it's
The lasers we're seeing are 10 centimeters between them, so this allows us to measure the organisms we are seeing. En pantalla tenemos los láseres que usamos para hacer una estimación del tamaño de los organismos que vamos observando. La distancia entre los dos puntos verdes que estamos observando es de 10 centímetros, así que esta tiene pues tal vez unos 25 aproximadamente. Unos 25 centímetros de longitud. Me gustaría enviar aquí un saludo a Leonardo Chacón, que nos ha estado siguiendo la expedición, ayudándonos con la identificación de equinodermos, de pepinos, estrellas de mar. Mucho de lo que conocemos de aguas profundas es gracias al trabajo de Leonardo y de Juan José Alvarado, los dos de la Universidad de Costa Rica, Museo de Zoología. Gracias, Leo, pura vida.
Yes. Yeah, copy. Thank you, Gage.
The green or the red?
The yellow dots.
Oh, we're almost there.
Yeah, that's right.
No se conoce mucho sobre estos nidarios que estábamos observando, hidroides, sin embargo, pues sabemos que son carnívoros. Atrapan pues, pequeños organismos como zooplancton, pequeños crustáceos, inclusive podrían atrapar pequeños peces. En pantalla tenemos otro ofídido o brotula. We were seeing a ophidiform fish. They are usually called brotulas or cuscils. Those are really important predators in these waters. One species of this same group was found on the Mariana Trench at more than 8,000 meters. A genus called Abisobrotula, really nice name for a fish living in deep waters. Now we have a sea urchin. Como pueden ver, los equinodermos son uno de los grupos más importantes que hemos estado observando. Ahorita estamos muy cerca de llegar a nuestro próximo punto de muestreo, número 2, de una serie de puntos que tenemos hoy planificados en un transecto de alrededor de 1 kilómetro, explorando este sitio al que aún no hemos puesto nombre. Hay muchos sitios que estamos explorando que aún no tienen nombre, que esperamos después de la expedición hacer todos los trámites para ponerles nombre y ponerles ojalá nombres en español. Okay, vamos a intentar acá con lo que llamamos Excalibur, una espada que SuBastian tiene para saber la profundidad del sedimento y saber si es la adecuada para no dañar el sensor y que tenga la profundidad adecuada para que todos los puntos de muestreo que tiene el sensor logren detectar las señales que nuestro equipo de investigación está buscando para hacer las mediciones de flujo de calor. Sí, lo que estamos encontrando es que esos sedimentos son tienen una capa muy compacta, muy dura de organismos calcáreos.
Yeah, even less this time.
Okay.
Y, okay. Vamos a movernos hacia el siguiente punto. As you can see, the sediment here is really white. This is called a calcareous ooze. I'm not sure if I'm pronouncing it right, but this basically is calcareous organisms that live in the plankton and then fall down to the bottom of the ocean as part of the sea snow. We have geologists here on board that specialize in this group of organisms. La razón por la que el Excalibur no está logrando penetrar lo suficiente en el sedimento, es porque tenemos un tipo de lo que se llama barro calcáreo, por eso lo vemos de color blanco, por carbonato de calcio, y esto es básicamente una acumulación de muchísimos años de lluvia pelágica, de organismos que viven en el plancton, que tienen estructuras duras en sus cuerpos, por ejemplo, foraminíferos principalmente. Conforme va pasando el tiempo, esos cuando mueren caen lentamente hacia el fondo marino y se acumulan en sitios como este. Así que bueno, parece que no hemos tenido suerte con las mediciones de flujo de calor. Esperamos encontrar algún punto que nos permita hacerlo, ya que sería bastante importante tener estos datos y saber si este es un posible sitio de carga o descarga de agua hacia o desde la corteza terrestre.
Maybe it's something that we have no idea what's going to show. So
Like, it can't be another dive
yeah,
after this, it's just focused on getting those, maybe that's possible.
I don't know. She asked for something like that. Yeah.
Cada uno de nuestros puntos de muestreo tiene 100 metros de distancia entre ellos. Vamos avanzando despacio, no solo para poder observar con detalle el fondo y poder hacer tomas de los organismos que observamos con detalle, sino también porque el barco, el Falcor 2, tiene que irse moviendo con el ROV, que está conectado por un cable hacia el barco. Así que es increíble toda la operación que realiza la tripulación, los pilotos del ROV.
With the pro, stabbing it and sliding it.
For those who cannot join yesterday's dive, we found a really big aggregation of skate eggs, deep sea skates. That was pretty amazing. We were able to take a couple of those eggs from the skates, and we found that they are viable. So we are really excited about it. We believe that those belong to the Pacific white skate species, which is one of the deepest elasmobranchs. Pretty interesting also because they have long incubation periods. There are studies available from other species that indicate that they can last between 3 to 4 years to develop. So that place at the Tengo Set Seamount where we were yesterday was really important. That we believe it's a nursery site for the species. We're seeing an octocoral here. Have you collected some octocorals?
Oh, I think I mean, we can maybe pick up that. Yeah.
I will ask Oda.
It is on the.
Can we stop for a bit just to ask if the researcher is interested in the hotel?
Of course.
I'm asking.
Sounds like maybe we might want to go up a bit check.
So with the eggs, we dissect them. We took some samples for do genetic analysis and see if we can be sure about the species identification. And we were trying to show that those eggs are actually viable. So that was pretty interesting. There are some studies, for example, in Galapagos and other places around the world that have shown that venting sites are important to increase the speed of the development. Yesterday we did not found that in that exact place, but we believe that the rays or the skates know something that we don't. So yeah, that we will try to come back and see if we can have a little bit more of information. I think we can go.
We can go.
Yeah, probably we will see more of those. Yeah, thank you. Lo que estaba comentando ahora en inglés es que ayer, para quienes se perdieron la inmersión del SuBastian en el monte submarino Tengo Set. Oh, sorry, they are informing that we have some pilot whales on the port side of the stern port side of the boat. So hopefully someone can have a video of that. Sí, lo que comentaba era que ayer en el buceo de Tengo Set, en el monte submarino Tengo Set, cerca de la cima, encontramos un sitio importante para las rayas de profundidad. Creemos que se trata de la especie, la raya blanca del Pacífico. Tuvimos la oportunidad de tomar un par de muestras de sus huevos o cápsulas y queríamos demostrar que estas eran viables, así que trajimos un par a bordo, las disectamos y encontramos, bueno, estaban en estadios de desarrollo diferente, uno más temprano que el otro, aunque los dos estaban en estadios bastante tempranos. En una ya podíamos observar el embrión. Estas muestras tomamos un pequeño pedacito para hacer genética, ver si podemos corroborar la especie, aportar un poco más al conocimiento de esta especie de la que se sabe sumamente poco. La especie a la que creemos que pertenece, Batirraja espinosísima, es una de las rayas que logra vivir a mayor profundidad en el mundo y además se cree que es una de las especies que tiene mayores periodos de incubación. Hay estudios en especies similares que indican que el tiempo de desarrollo dentro de la cápsula puede variar entre 3 a 4 años, así que es bastante lento, sumado a que son especies que suelen tardar mucho para llegar a una edad madura. Así que es una especie pues, que la hace bastante vulnerable por todas estas características de su lento crecimiento y edad muy tardía de maduración. Así que estamos emocionados por el descubrimiento que hicimos. Además, encontramos señales también de emisión de agua desde la corteza. Una señal pues no tan fuerte como en Dorado, pero igualmente muy importante. Así que son algunos de los hallazgos más interesantes que hemos tenido durante la expedición.
Yeah. I don't understand. We said, looks like a baby, but I don't think that's how they do.
They're cuddling.
Yeah, the Caroline. Yeah, deep sea love.
It's cold down there.
Can we, just wait for a second? I think
Sure.
this
Okay.
might be a good collection opportunity, but I'm not sure. Yeah, let's do this one. Like, I didn't mean to.
This is
Can you take a screenshot
Yeah.
with the lasers? Yeah, that's right. That's why we think those two pukes.
That would have to go in the final box, I think.
Yeah.
It's the fun to try to get that one in a quiver.
So we have to put them in bio box number 3, right?
Are we for sure collecting this?
Yes.
Yes, please.
Yeah, so let's put it in 3A. Ideally, we get both of these in.
Hay interés.
You guys see the tentacle going now?
Can we zoom in before we?
It's eating. Yeah.
Not anymore. Oh, that, the white one?
It pulled. Yeah, look, it's pulling it out of its mouth.
Oh. And look, there's a small crab on the left.
Oh yeah, this is one crab there.
I don't think I saw it. This is before I watched it.
Oh, it's not.
Yeah.
Okay, anyway, sorry.
Thank you. We didn't see that. Ya que los equinodermos son uno de los grupos más importantes que hemos estado encontrando, hay un interés en recolectarlos para entenderlos mejor, no solo para poder identificarlos, sino también para estudiar su microbioma. Tenemos a bordo a Holly Lutz, que es una investigadora asociada al Field Museum de Chicago, que está analizando el microbioma, que es toda esa comunidad de organismos que viven dentro de los animales, virus, bacterias y demás, y que se cree cumplen funciones bastante importantes dentro del organismo, sobre todo en sus tractos digestivos. Así que es parte de las investigaciones multidisciplinarias que estamos haciendo. Si tienen preguntas, pueden dejarlas en el chat. Nos encantaría poder responderlas. Aprovecho también para invitarlos a visitar la página de Schmidt Ocean de la expedición. Ahí vamos a estar compartiendo blogs donde explicamos tanto en inglés como en español lo que estamos haciendo. So for those who are joining us in this moment, we are exploring a site offshore of Costa Rica without a name. We are exploring a lot of sites that doesn't have names yet. So it's part of what we want to do, to name all these sites and have more information of these unexplored sites. This is the first time we are exploring this location. Oh,
Yeah, I did the crab of the.
also, yeah.
The shrimp thing on.
So yeah, we have this month we have a lot of birthdays here on board. So we have a sign that we remain there for the whole expedition because there are many people celebrating their birthdays in these days. So yeah, happy birthday to J-Rod and also to Julie, one of the organizers of this expedition, Julie Hubert, that she's not here, but she's a really important part of the expedition. Como les hemos estado comentando, todas estas recolecciones que estamos haciendo de especímenes cuentan con todos los permisos necesarios del gobierno de Costa Rica y van a ser depositadas en el Museo de Zoología de la Universidad de Costa Rica, donde va a permitir a investigadores especialistas en los diferentes grupos a realizar aún más, a obtener mucha más información. Oh, no, no, no. Oh.
It's not too late.
Is there?
Might be able to fly down and get it up. We'll
Okay.
see.
Okay, it's not lost. Well,
Yeah, I'm just gonna try and press down and see if I can fly it.
okay.
Yeah, I don't know that you'll be able to grab it.
Okay.
So, that's why I'm
Vamos a intentar encontrar el pepino que se cayó del brazo. Esperamos que esté por ahí aún.
There it is.
We're seeing what's happening in this world.
Crash the seabed. Not coming out of there. I'm gonna have to put some progress. Come up again.
Walk a little bit, maybe I'll unlodging from where he's.
Yeah, let me get to the altitude again. That way I have room to come down.
Para quienes se unen en este momento, estamos explorando un sitio enfrente de las costas de Guanacaste, a unos 200 kilómetros aproximadamente de la costa. Oh, there is. Whoop. Oh, that was close. Uf. Estamos explorando una localidad sin nombre aún. Parte de las cosas que queremos hacer es lograr nombrar todos estos sitios que estamos explorando que aún no cuentan con un nombre.
Where did the ball go? It did go in the box?
I think he's actually like
Right inside of it. See that?
Oh,
Where my
he's
finger
inside?
is? But it's too
Yeah.
It's there, yeah.
I inside the box, yeah. Ooh.
following.
Yeah. Whoa. Wow.
Let's just let him land out or something. I don't know if you're gonna. I mean, I guess he could get a shot at it. He's awful small.
Whoa. Yeah, I think they will get the prize. These pilots are really amazing.
He's almost down. Let's just let him land out.
If you want to know more about the expedition in general, please visit the Schmidt Ocean website. We are uploading their blogs where all the scientists explain what they are doing. So it's a good way to understand a little bit more with more detail what we are doing.
What was crazy is the thrust of Urchin did the opposite effect.
I think that it landed, so we will be able to grab it.
I'm not steady enough. Why am I spinning?
Pull the fortune a little bit.
Tanto hoy como los próximos días vamos a estar teniendo turnos de 1 hora para que todo el equipo científico tenga la oportunidad de conversar con ustedes y explicarles el trabajo que estamos haciendo. Tenemos un equipo bastante grande de personas que hablamos español, así que es una buena oportunidad para entender en nuestro propio idioma lo que estamos observando. Así que invito a que se queden hasta el final. Hoy en esta exploración son las primeras imágenes que tenemos gracias al ROV SuBastian de las profundidades de Costa Rica en esta región. Cool. Yeah, that seems like a hot dog.
We're almost at a waypoint where not a lot of penetration to the scholar. So
Vent. Oh, yeah, not.
All right, we get it. Thank you. Pero van a aparecer más. Pues, ya lo tenemos, pero ahorita van a aparecer más. Es que no quería ponerme a colectar mucho.
Sí, no, porque ya no quiero, hasta que hagan todo eso.
Exacto. Yeah, they are together again. Until the end, yeah.
it's arrived on its waypoint. It's there. But it's
So we are getting to our waypoint 3. So soon we will be trying that again to measure the thickness of the sediment and see if we are able to make the heat flow measurement. Estamos cerca de nuestro punto de muestreo número 3. Así que vamos a estar intentando nuevamente. Como les comentaba, cada punto de muestreo tiene 100 metros de distancia entre sí. Y vamos a estar deteniéndonos esos cada 100 metros para intentar hacer una medida de flujo de calor. Hasta el momento no hemos tenido suerte porque el sedimento es bastante duro. Necesitamos una profundidad mínima para poder realizar estas mediciones. Okay, this seems like maybe a nice spot. Yeah, we go on to the end. Yeah, cool.
Yeah. That's, man, it's also, we have a smaller floor.
And it's going straight.
We'll try again. Yeah, exactly. This pokey stick is, we call it Excalibur. Is checking how deep the sediment is to check if we can put in the heat flow sensor that will be measuring temperature, and that will give us evidence if this is a discharge or charge site according to how low or high the temperature in the sediment is.
Yeah, let's keep going.
Yeah.
Rather not.
Okay. So our team decided that it's not deep enough, so we will continue to our next waypoint. En este caso, la profundidad del sedimento no fue suficiente para poder penetrar el sensor de temperatura que se utiliza para medir el flujo de calor. Así que vamos a, vamos a seguir avanzando hacia nuestro siguiente punto. Como les comentaba, una de las razones por las que estamos teniendo, pues digamos, inconvenientes para hacer estas mediciones es que el sedimento es bastante duro a causa de una gran cantidad de organismos que en algún momento fueron parte del fitoplancton, que tienen partes calcáreas en sus cuerpos. Con el paso del tiempo, conforme mueren, se van depositando en el fondo marino, como lo que se conoce en inglés como marine snow o en español como lluvia pelágica. Como les hemos estado comentando, esta es una zona de alta productividad cerca del domo. Estamos bajo la influencia del domo térmico de Costa Rica, no exactamente en el domo, pero bastante cerca. Así que tenemos muchísimo plancton y esa es una de las razones por las que tenemos este tipo de fondos tan ricos en materia orgánica y tan ricos en carbonato de calcio al mismo tiempo.
Updated waypoint group.
Copy.
Hoy estamos justo en la mitad de nuestra expedición, así que aún nos quedan muchos descubrimientos por hacer. Hasta el momento, entre algunos de los logros más importantes de la expedición está el estudio de los pulpos en el Dorado Outcrop. Fue nuestra primera inmersión, vamos a volver a ese sitio, ya que dejamos algunos experimentos ahí. También hemos observado pulpos en otros sitios que nunca habían sido explorados. Hemos estado caracterizando toda la fauna que observamos. Y por supuesto buscando evidencia de estas salidas de agua más calientes desde la corteza terrestre, que es uno de los aspectos que creemos muy importantes para el desarrollo de los pulpos. Today and the rest of the days of the expedition, we will be having both English and Spanish narrations of our dives. So we will be having shifts of 1 hour each. So you are more than welcome to join us and ask us questions in both languages. Half of the science party is from Latin America. So as a Latin American, I think that's really neat. It's a great opportunity for us to be here. We don't have that much opportunities here in Latin America to do this kind of science with all this equipment and technology. So we, the Costa Rican and Latin American members of the science party, are really happy to be here, really compromised to do our jobs well. So we are really grateful for this opportunity. Another cosquil, una brotula. Como les comentaba, una de las familias más importantes en los peces de profundidad. Este es un gusano escamoso. Mide cerca de 10 centímetros probablemente, como para los que no se vienen uniendo y no saben que son estos puntitos verdes que estamos observando. Es una escala que utilizamos para estimar la medida de los organismos. Este gusano que estamos observando es un poliqueto. Está emparentado con las lombrices, del mismo grupo al menos, de los anélidos. Es un gusano escamoso. So we are halfway to our next waypoint number 4. We still haven't been able to do the heat flow measurements. We have here in the control room the heat flow team. So hopefully they will be able to do their jobs. We've seen some we are seeing some octocorals and bryozoans. As well of brittle stars, of course. Como les comentaba, si quieren conocer más sobre la expedición, pueden visitar la página de Schmidt Ocean. Ahí tenemos una página exclusiva para la expedición y ahí estamos subiendo blogs que diferentes personas del equipo científico escriben para tener la oportunidad de contarles con más detalle todo el trabajo que se está realizando a bordo. Todos los blogs están tanto en español como en inglés. Hasta el momento hay 2 en línea de una serie que vamos a estar subiendo. Hoy estamos justo a la mitad de la expedición, así que aún queda mucho trabajo a bordo por hacer. Another orthocoral, they usually have stars living up in the octocoral. Nos estamos acercando al pilot whales.
Pilot whales.
Okay, that's why Levi is not here.
Yeah.
Oh, no, I'm okay.
I was
I think Alex.
Yeah. I think this will
Nos informan que tenemos un grupo de ballenas piloto. Así que gran parte del equipo está afuera viendo las ballenas mientras nosotros observamos la maravilla del fondo marino.
Look, Carlos, this is the best I can do. You know now who will help us.
Yeah, we haven't seen sharks so far, but we have seen skates, really beautiful skates, Pacific white skates, but no sharks so far.
I've been noting the depth that it's got over those, and then whether. Oh, thank you. Right, actually.
We haven't gotten any yet.
Okay, and then we just use that.
I'm gonna go.
Hola, Miguel. Buenos días. We have a group of pilot whales close to the Falkor too. So most of the science party is there. This is an important area in the pelagic waters for whales and other migratory species. We are in the area of influence of the Costa Rican thermal dome, a really important area for migratory species like big whales. Hi, Diva. We are between waypoint 3 and 4. We haven't been able to do the heat flow measurements because the sediment is too hard. It's full of, this is what Maria called calcareous ooze.
So no
So,
heat flow yet.
Not yet, yeah.
And one sample, which was the sea cucumber.
2 sea cucumbers that were
Did
together.
we get? Yeah, I was about to say, did we get both? Okay.
They were like.
But then one like fell. Yeah.
Yeah, but we were able to.
Right.
Yeah, and that's it. And before they catch another sea cucumber, so we have 3 sea cucumbers.
Oh, okay.
Yeah, this is together here in the shoe.
Perfect.
Here's one really big. Para quienes nos preguntan si estamos abajo en el submarino, no, nosotros estamos en el barco, estamos trabajando con un robot que está conectado por un cable, así que no hay nadie dentro del robot. Es una de las ventajas de trabajar con robots. Bueno, una de las ventajas de trabajar con robots es que tienen periodos de trabajo mucho más largos. Son capaces de estar bajo el agua por tiempos bastante prolongados. Y a través del cable se transmite toda la información que estamos viendo con todos los sensores que tiene el robot de temperatura, oxígeno, tiene cajas donde podemos poner los organismos, tiene 2 brazos que permiten realizar colectas y hacer diferentes trabajos bajo el agua. Y cuando el robot regresa, pues podemos analizar todo lo que trae desde allá abajo.
Thank you.
El robot tiene una serie de cámaras, así que tenemos, lo que mostramos ahorita es la cámara principal en la transmisión, pero además tiene otras cámaras que nos permiten monitorear el robot y otras partes podemos observar, por ejemplo, algunos recipientes donde se colocan muestras. Así que son bastantes cámaras, mucha tecnología de punta. So right now I will give my space in the seat leading the dive to Diva Amon. She's from the Caribbean, so has a lot of knowledge about the deep sea biodiversity, lots of experience. So good morning, it was a pleasure to join you this morning, and hopefully you will stay with us for the entire dive. Muchas gracias. En este momento voy a ceder mi espacio en la silla aquí liderando el buceo a Diva Amon. Ella es una bióloga de Trinidad y Tobago con mucha experiencia en el estudio de ambientes de profundidad. Conoce muchísimo sobre la biodiversidad de este tipo de ambientes. También vamos a tener un cambio en el equipo de pilotos, así que por unos minutos vamos a estar haciendo este cambio. Muchísimas gracias, buenos días, espero que nos acompañen el resto de la inmersión y de la expedición Pura Vida.
No, I went out like way early and they were not there, and then they have reappeared. She goes, they're still there. Go see.
So far we haven't been.
I was excited.
So good morning, everyone.
My name is Dr. Steve Amon. I am at the University of California, Santa Barbara in the Benioff Off Ocean Science Lab, but also with Species, an NGO in Trinidad and Tobago. And I'm joined in the science party by Dr. Miguel Semedo from the University of Porto in Portugal. And we're going to be your science team for the next hour. How was your vacation?
It was wonderful.
Happy birthday, by the way. So we actually, I'm not sure if this has been said already, but we actually have 3 birthdays on in the Octopus Odyssey team today. J-Rod, our pilot, who's on right now. Carl, our pilot, who he just took over from. And Dr. Julie Huber, who is one of our scientists on shore and one of the principal investigators of this grant. And it's also apparently Jacques Cousteau's birthday. So, you know, ocean exploration all around. So we are essentially following a series of waypoints this morning on an unnamed outcrop, which we're just saying is outcrop E. No one has been here before. We are the first to map it. And so we now have some pretty good bathymetry. And we are essentially trying to get some heat flow measurements. And so far, we are, ooh.
Keep going.
Yep.
Yes.
Okay.
So you can probably hear some audible yes coming from the room. That's because we are, I think, at waypoint 4 now. And yes, waypoint 4. And so far, we're supposed to be taking heat flow measurements at every waypoint to try to understand what is happening subsurface and whether water is likely to be discharging or recharging and whether this area is hydrothermally active. But we have not been able to because actually the sediment has been too shallow. So this is the first spot where we're going to be able to get heat flow, which is really exciting. And yeah.
No. What we really don't want is tell them. Yeah, we're sorry.
You're like a kid.
Any place in particular, Ben?
No, just even, 10 centimeters away from where you put it. Yeah, wherever it's fine.
So we're just doing a few tests here to make sure this area is in fact has deep enough sediment. And we'll get them to flick the bathymetry, the zoomed out bathymetry up at some point. But where we're essentially going is from one high point on the seamount to another, but traversing through what could be like essentially a sediment, a low sediment pool, if you will. So this may be that we're now deep enough. Sorry, yes, deep enough. That's correct. Down the hill and hitting the more sedimented areas. Good. Just this is good. They're working. Good. Any wisdoms you'd like to impart?
You want to go see the whales?
No.
Okay.
Thanks though.
Okay.
I saw pilot whales in March. It's fine.
Yeah.
So there's a lot of excitement on board because there is a pod of pilot whales off to the port side. No, maybe now it's starboard side.
No, they're still on the
Some
star and port side.
Okay. Yes, so there's a lot of excitement on board. All of the scientists are outside and many of the ship's crew. Do you know how many?
I think at least 8.
Woo! At least 8, Beth is telling us.
Yeah, they started coming up with their heads first.
Oh, the spy hopping.
Yes.
Oh.
Like, I think they're checking us.
Yeah, 100%. So wait, when we went out at first, where were they? Too far, maybe. Right, okay.
Which means they've been following us because we've been moving away from them.
Yeah, they were here when I went for a run at 5:30 this morning. They were circling up.
Where did you go? The local park?
Oh, yeah.
Oh, and they were there already, Scott?
They were there during lunch.
Oh.
We're doing the bottom part right now.
We're doing it right now, though, so it's fine.
We'll put my skid where that.
Oh, yeah, you're right. We need to hop up, because where it's disturbed, like,
Okay.
Okay. That's still fine.
Yeah.
Yeah. Yeah.
Otherwise, this will just be a
Yeah.
calibration file.
Oh, that's water thing. Oh.
Right. We're good to go in there?
Yeah. There was a bunch of stuff.
Did you see them, Alice? No. And that's a reminder. I'm sure many of the scientists are going to be posting the pilot whales and many other amazing things. So do follow our hashtag on social media, hashtag Octo Odyssey, where lots of us are posting bits from our daily life, the samples we're collecting, cool things we've seen on the seafloor. Probably a best way to get a behind the scenes look.
All right. Are you ready for it to put it in, or are you still doing background?
We have 30 seconds.
Okay, we got a clean spot over there. Not using a lot of thrust at all.
We're ready to go when you are.
Roger that. No doubt.
Alex, there's a question how big the outcrop is. Do you know?
I can pull up the map here.
Yeah, there's like this map is just showing, it shows basically no contours with measurements, no scale.
Yeah, sorry. I just got sat down here. Let me
Yeah, take your time.
let me look it up for you though.
We have time. We're in the same spot. For the
See all the way down. It looks like maybe the fifth thermostat is probably not going to be in.
Yeah, 5th is not in.
Pull your arm up a little bit. Just a second.
Yeah, we're just trying to figure out the size up.
You got 3 out of 5?
Yeah.
All right, I'm going down right now.
Yeah.
4 out of 5.
Okay.
Yep.
Yeah.
Yeah, we got a response.
Is that all signed?
Yeah.
After lunch, I'm gonna come see.
I can do it again if y'all want. I
Fine, you're good.
think I just need to have a little bit more vert to start off.
I think we're good. Yeah. Yeah, we got them all in. Thank you. Thank you.
How long do these sessions take?
Takes about this point, like 13 minutes or so, 14 minutes.
So, if you haven't joined us before, we're doing some heat flow measurements, and from when the probe goes into the sediment, it takes about 13 minutes. So we're going to be in this exact position for that amount of time, which means we're going to be doing things like this, taking some good zooms at the seafloor around us, getting to know it really well. So this is a brittle star or ophiuroid. The Eastern Tropical Pacific is actually the abyssal areas of the Eastern Tropical Pacific, like where we are known to have remarkable biodiversity of brittle stars. Though, as you can see, they're really hard to ID because they're often covered up with sediment, or you can't see many of the distinguishing features. So yeah, something we've been seeing a lot of on our dives. Can we take a look at that stick behind?
Looks like the main outcrop here is about like a kilometer across at the top, like this top little
Okay,
bulgy
okay.
area, but then the whole area maybe 3 kilometers across. There's kind of like a tiered step up.
Wait, can you put the, oh, it's up. So for those who were asking how big this outcrop is, Alex has just flicked up the
Oh.
bathymetry.
We'll flip.
Oh,
There we
will
go.
flick up. Sorry. Here we go.
You're good, you're good. We're just cutting across this little area here.
Right. Yeah. So as you can see, we're heading down into that little depression, which is why the sediment is getting thicker. Oh, another brittle star. Great. Oh, many brittle stars. Can see 3 in shot right now. Let's take a look at if we can. Oh, hold on. Broken the pointy thing. That. Please. Oh, a little bryozoan. So life on the abyssal seafloor tends to be quite small. So it's only when you take a much closer look, like what we're doing now, that actually a lot becomes clear. So here we can see we've got a little bryozoan. On the right of it are 2 xenophyrophores that basically just look like muck. But each of those structures that you can see there is a single cell that creates these giant tests or structures. They're called xenophyrophores. They're basically giant foraminifera. And on a previous expedition to Costa Rica, also with with Schmidt Ocean Institute, but on the first Falcor, they actually found that these xenophyrophores can be used we knew they could be used like we're seeing with the ophiuroid there, where the ophiuroid is using it to attach to, shelter within. But we also found that, colleagues also found that fish were actually laying their eggs in them, deep sea fish. And so there is some kind of they also play a role, of being a nursery. Yeah, can we look at this? Because I think it's alive. It's got a track. So this also looks like muck, but if we zoom out a little bit, we can see that there is actually a track behind it. So like it's kind of like hauling itself along the seafloor. So maybe a tubed polychaete that's built its tube from the surrounding sediment and then just like carries its whole house with it. I don't know. Oh yeah, yeah. See, so I'm not imagining it. Yeah. Yeah, so it's definitely moving along. Can we go take a look at it again, see if we can see anything like a little head poking out? And we'll come back to that coral thing. So judging by the trail, this looks like it should be the back end, unless it walks back to front. Yeah, right? Weird. The deep sea is weird, y'all. Weird but great. Weird but wonderful. Yeah, so something super small there. Either an anemone or like a corallimorph or Sarianthid, some kind of cnidarian. A teensy little one right there. Gosh, how big is that's like what? A few millimeters? Wow. Sure. While we're here. Lots of xenos. There's there's actually a good few. So this is a great example, a better example of xenos, and they can have two different forms generally, plate-like forms and reticulate forms. And so this is an example of a plate-like form because, as you can see, it looks like it's kind of composed of different plates. And the reticulate forms kind of look like little nests or sponges, like little brown sponges. And yeah, then there's this thing. Yeah. Oh, can't see the base. I think you're right. Yeah. No, right? A little baby sea pen. Where is it on this? Here, point. Oh, yeah, okay. Yes, it is really tiny. How are we doing back there, Seafloor team?
It's pulse. It's looking good.
Okay.
How's the noise?
It's like really little. So I don't think we'd be able to collect it properly. Yeah, definitely we see a bigger one, but this one's like really small. Oh, yeah. What else we got? Oh, look, another one. Up in the top, right? Unless that's just trash. Do not know. Yeah, like it literally looks like something built a little sediment nest. Yep, no idea what this is. But all of these things are really good examples of bioturbation, which is essentially all of these kinds of marks you can see on the seafloor, tubes, poop, burrows, whatever. All of that is basically deep sea life going about its day-to-day living, but at the same time working to bury carbon down in the deep sea. It's really important for carbon sequestration. Oh, is that another one? Oh, it's okay. Yeah. Don't know what, never seen that before, but weird. There was another stick thing. Yeah, up there. Mm-hmm.
Looks like there might be something sticking out the right side very slightly.
Very small. Yeah, little white thing. Oh yeah, that's definitely something else alive. Yeah, that might be a piece of it. Maybe not. So we are quite zoomed in now, which means that you often would not be able to see the lasers, but we can flick them on just so everyone can get a sense of scale. I mean, these are some really tight zooms. So some of these animals are quite small. There you go. So you can see the 10 centimeters there. Yeah, lots of xenophyophores around. Yeah, and what's this? It's like, you know, when you start to see like shapes in clouds, and that's what we're doing right now. Oh, that looks like a polychaete. Do you have any more zoom? It looks like we can see like chaete sticking out.
Yeah.
Like there. There's also a little ophiuroid. Yeah, I think the little yellow thing is probably a sponge, but then you can definitely see like strands sticking out that look similar. Oh, I was gonna say like you when you're on your holiday, you missed. We kept finding sea mice. So they're like the super bristly polychaetes that are like, they were like gold. Like they literally were like the most fabulous thing we've seen all expedition. They just were like, if you took carnival and put it into an animal, that's what it would be. So that's kind of like what those chaete look like sticking out. So I'm caveating all this by saying I'm not a geologist, but I suspect that when we have been pulling the probe out of the seafloor, it's been this is from disturbed below, and essentially that's the calcareous silicious ooze that we've been seeing forming layers of the seafloor in past dives.
Gotta love oozes.
People, how do you feel about that statement?
Oh, just oozes. Yeah.
Harder under crust that we've noticed, but it was softer here, so, but it seems to be everywhere, not just where we are right now. He's hiding.
I wonder how much they move. Like, has he been there for like 5 months?
It's kind of a weird little catch-22, right? I mean, if you move, you burn energy.
Mm-hmm.
But if you're just sitting still in the wrong spot, you might not be getting any energy anyway. So.
Mm-hmm. Yep.
The strategies of the deep sea.
Oh, a teensy little amphipod crustacean living on the side of that xenophyophore. And so there's a lot more life than we're actually seeing right now, but it's just living within the sediment. So the majority of life on the abyssal seafloor is actually found within the top 10 centimeters of the sediment. And many of them are very small, but there's very, very, very high diversity. What else we got? What about over there?
I think we're ready, done with
Oh.
the people measurement.
Wow, was that 13 minutes? Wow, time flies when you're having fun, guys. So we are at the end of our heat flow measurement. So we're going to retrieve the probe and then carry on to waypoint 5. Please.
Sorry.
It just, I think coming up out of the silt, it wouldn't make that very satisfying, like shing of drawing it out. But I imagine it every time.
I told the other pilots that it's the way to stop. I'm basically telling myself that I've done it.
Oh no.
Okay, and away we go. Is it possible to fly either closer to the seafloor or just zoom more? Just, yeah, once you're situated and settled.
Hey, on the bridge, got an updated waypoint for you.
So a good few of these pencil urchins hanging about, Sidarids. Haven't seen many of those on previous dives. Also Relicanthus, which is a relative of the anemone. And 2 fish. No, 1 fish and 1 Cyclopodes. Sea cucumber. Well, it's a big Cyclopodes. This looks like a
Yeah.
Synaphobranchid, maybe. Maybe we can get a quick zoom at the Sidarid and the fish. Let's do the fish first since it's mobile. And then the pencil urchin. So right there we can see 3 different types of 3 different classes of echinoderm. Urchins, a brittle star, which is the middle one, and then a brisingid. Oh, maybe that's an orphirid. Yep, and then we have a synaphobranchid, which is a cutthroat eel, is the common name. I'm always amazed how many scratch marks they have. Like, what have they been doing down here, really?
Hurry, ROV. Yeah, we got an updated waypoint for you there.
That's great, thank you. Great shot. My pectoral fins are so small. Great shot. Sure. Or we can just continue. I'm sure we'll see more. Oh yeah, there we go. It's right there. I'm not sure if we've gotten any close-up views of these. But yeah, these are common name pencil urchins. And they've got much thicker spikes than others and can get even thicker than this. Cool. Great, thank you. We can continue going. Then we've got a Cyclopodes with a giant sail. Like possibly the biggest seal I've ever seen on one of these. Oh no, or is it half standing up? Yeah. It's doing its morning yoga. Oh yeah, it is half standing up. Weird. It's a sea cucumber.
Reaching for the sky.
Oh, maybe it's gonna swim. It's definitely lifting off the seafloor more. Oh, and you can see it has a little something living on it. Polychaete, crustacean. Hard to see from here. Get a good look at all of its tube feet. Its mouth.
It's just figured out how to turn around quickly as a sea cucumber. That's all. You know.
Yeah, sounds great. Oh,
Cukes are great.
oh. It's a good looking cuke, I must say.
Yeah. Yeah.
Alrighty.
Got like a full duckbill kind of going there.
Yeah. And it just
Duckbill
looked like
lobster.
really like clean and
Yeah.
solid. What is that? It's fine. I mean, we could just do a quick zoom, but like just a quick one, then we'll go. I think it's actually just a sea cucumber.
I think it's
Just
one
a
of
sea
those,
cucumber.
like,
I feel
spiky
bad to say
cukes,
that.
Yeah.
Yep. Wow, look at this little buddy. Haven't seen one like this before. I don't think I've ever seen one like this before, actually. This is like an Onerophanta type thing, but purple. Maybe it's a young one of a... Interesting. Okay, we should move it along before Sergio comes running in and says, we need to collect this now.
Sorry.
Thank you, Carla. That was great. That was a weird one. So one of us explains that the main animals the main species or taxonomic groups tend to be the echinoderms, so brittle stars, sea stars, and sea cucumbers. You can see a ophiuroid on the move over there, as well as xenophyophores.
Looks like we got a
whole
little.
bunch of like cukes we've not seen before. Interesting.
There's some
Was
sediment
this
turnover.
us?
Yeah.
This we didn't do this. CTD? Did we do a CTD here?
No.
Oh, can we have a look to see if it's like a burrow? So I think there's been a few theories said about what these things could be. Are we sure that's not us? Okay.
According to my snail trail, I've never been.
So there's a few different. So Janet was saying there's some squid that copulate close to the sea floor and then create marks. It's known that deep diving whales sometimes come down to the seafloor and then essentially run along the seafloor, gouging, running their lower jaw into the sediment and causing these sort of gouge scars. But yeah, this one's a weird one. Interesting. Cool.
Yeah, there was another one. Or more disturbance over there.
Yeah, it could be an animal making a burrow. Could be from geological forces.
I mean, this is old enough that there's something settled on the debris over to the side
Good
there.
point. Yes.
I mean, it's something fast, but relatively speaking. Oh yeah, there's a couple stars down in the debris. So
But I think you're right. The sedimentation rate is like there is sediment down there. So who knows how long these things might have been here. Cool. And yeah, some brittle stars in between. Coolio.
That was a very warm spot.
Was it?
Yes.
Wait, where were you heat flowing?
Yeah.
oh, okay.
Oh, so heat flow is telling us that was very warm. Wait, at this previous measurements indicated at
Over
this
the
site.
side, they were over the side measurements, so it's not clear how accurate their placement was, but they said about 120 milliwatts. We got over two, we got more like 2.
So what does that compare to like Dorado?
It's not as warm as Dorado, but it's definitely warm enough to be interesting. Over 200 is kind of.
What was yesterday?
Yes, we didn't get any measurements for 2 days.
130,
Yeah.
Wait, what did you say? Wait, what?
130, 140. It's 5 days.
The
Did we
baby
see any
one
outflow
thing.
there?
We only did
We haven't
one
scanned it out,
transect.
though.
Yeah.
Right.
So we didn't really spend enough time there, but we would expect for this crust age to be 110. So we're towards
Okay.
that.
Interesting. Cute little hard substrate here. Good example of the fact that hard substrate is in short supply. And that means that when it does exist, lots of life grows on it and uses it. So here we've got a sea star, a brittle star, a sea spider, a coral, and perhaps a carnivorous sponge. And another sponge. No, I think the other one is not a carnivorous sponge. It's a coral. So 2 corals, a serpulid worm, 2 different types of worms, 3 different types of worms, a sea spider, a sea star, and an ophiord. All on this little bit, which is like, what, 10 centimeters more, less, 20 centimeters across? Yeah. Cool.
Oh,
Whoa. A Munidopsid isopod, maybe? Yes.
that's a big one.
That is a really big one. I don't think I've ever seen one that big, except if it was swimming, maybe. But so this is an isopod, a type of crustacean, and sometimes we see these swimming with their legs sort of flailing around them. But yeah, could easily be confused with a sea spider. Cool.
Missing a limb, it looks like. Oh, no. I was gonna say there's another, but that's one of those pencil urchins.
Oh no, there was another one just to walk just below us.
Oh, okay. Yeah. You ever seen them swim? That's fun.
Can we have a quick zoom on this? And flick the lasers on so we get a sense of size. I think ultimately we are going to want to collect one of these at some point, but we want it to be a size, like a not too small, not too big one. This one's only about 10 centimeters, so let's leave it.
I don't know if we have
Yeah, you're right. Definitely a sea fan. With a mysid living on it and a sea star. No, that's fine. We'll collect one later on. Let's put that as a priority.
where waypoint 5 is going to be.
Okay, great.
We're going to be able to do a heat flow. There, might be clear.
Oh, yeah. Rach and Rob, waypoint 5 might be on a little outcrop, so we're just gonna look around and find a good spot.
That's, we're opportunistic.
Ooh, look at this. Can we do some quick zooms? And also there's a fish, I think, behind. So there, I think there's a fish up there, but we'll get to it. But ooh, an interesting little outcrop here where we can see some of these encrusted lobate lavas. And you can see immediately how the abundance and diversity goes up, right? Suddenly we're seeing a lot of these teensy little corals, sponges, sea stars.
There was a worm with its feeding appendage out from under a rock down there.
Yeah, a lot happening in this little spot.
Yeah, it was over on the right. Back a smidge more, I think. Right there. See the white sticking out from under the rock there.
Oh, good spot.
Oh, it's gonna get back in there.
The little,
Just to the left, the
little tubey
blazers.
next to the
He's
pink.
slowly pulling in. Yeah. Yeah, by that
Yes.
sea star. Pulled in more, but
Some
yeah.
stalk sponges, worms, lots of corals, brittle stars.
It is cool though. Like you get that change in bottom,
100%.
and then immediately change in the life that's present.
Yep. So I think let's maybe find a spot off to the right to do the heat flow, and then we can come back and collect one of these tiny little corals, please. Looks like there's a lot of soft bottom, hopefully, around here.
I'll try to fix this area.
Yeah, that sounds great.
All right, we're looking for another location for heat flow.
So we're just setting up to a heat flow, okay? Yep. Another fish, another one of those chewy things. Oh, and another one of the sea pens. So we could almost do like two collections. Oh yeah, one of the weird yeah. So here we can see we've seen a few of these on this expedition. It's like basically bioturbation in a perfect ring of holes.
Yeah.
Some animal that lives within the sediment creating that. Okay, so we're at waypoint 5 and we're trying to do a heat flow at every waypoint. So we're going to try for our second heat flow here because we haven't been able to do them at some of the earliest waypoints on this dive because the seafloor has been too hard.
Are we ready?
Go, 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 go. Yay!
Are you happy with that, or are you going to want to do another stab?
No, I think that's good
All right.
stuff.
Let me see if I can. Go down just a little bit deeper now, then I'll put it down a little bit lower. All right, pretty there. Now I'm going to use up in the time.
Hmm. Oh. I didn't think the sediment cores were like even on the.
That means stable, the sediment's not disrupted right there.
Just let me
Good
ask.
for me to go down?
Yeah, let's ask.
Okay, just a sec.
Good call, good call, good call.
Okay, recording, we're ready.
Ready.
Okay, rope going down.
So there you can see the probe descending into the sediment.
Should be all 5.
Or last one's not.
There you go. Now, how about now?
What's that?
How about now? Still no?
No heating on number 5.
Actually, there is. It's, yeah, it's.
I just got it to go down like another.
Yeah,
Okay.
it's just minor.
Okay.
Yeah, it's there. Yeah, it's.
Oh,
Sorry. 5:35.
okay. So there is a chance that if Rachel tells us the heat flow is elevated here, then we will do sediment cores also. Just getting up the fish guide to see what this dude is.
Can you?
So I think this is a cusk eel,
quick shot
family
of the
Ophidiid.
60, and you might need to drop
Could
it.
have been like a Barothrites, maybe.
Yeah, drop it down now. One more. Yeah, I'm just trying to get rid of those bars.
Well, I'll get rid of them.
Oh, okay.
So we've got we've seen quite a few of these on this expedition, these tubed worms that hang themselves out of the end of the tube. Down here, we've got one of these sea pens. I don't know whether we're able to like sample while we're doing this. No, probably not. No. Well, this could be a good sample to collect when you're done. Ooh. So yeah, we might be here for a little while, collecting some animals, collecting some push cores, and of course doing our heat flow right now. But this looks like a brittle star. This is one of the brittle stars that does not live on the seafloor. It hauls itself up onto structures to get itself as far up off the seafloor as possible to then extend its arms, which you can see have got those little structures coming off of them that it uses to sort of filter the water and get food. And it looks like it's living on a sea pen, which perhaps is too small for it. Pelagic. Ooh, gorgeous. It's got like almost like a flower, what looks like a flower coming out of the end of the... Okay, so I think there's the Pelagic tube, then the worm itself hanging out the end, and then all of these little things living on the tube. I think are carnivorous sponges, judging by the structure. No, I think they're carnivorous sponges. Do we have any more zoom? They look like they've got. Oh, and there's another polychaete living on it. A scale worm. Here. You can see all its little scales still on. And its head. Wow, this is great. Yes. So I'm. Oh, I don't know if those are those hydroids. Oh, I don't know. No, I think you're right. So they have like, because they've got structures going up the stalk and then at the end. Okay, weird. But the scale worm is really cool. So this is from the family Polynoid. And you can see they've got literal scales that cover their back as a form of protection. And when they're collected, they often drop those scales, sadly. Cool. And it's, yeah, hanging on for dear life, looks like.
What is this dog animal called?
The big brown stalk?
Hey, Pete, you got me? Pete, you got me?
Yeah, I did.
Oh, okay. You're on RV. Yeah, if you could come to the top there, that'd be great.
Yeah, it's good to RV. This bridge is built as like a real meta.
You want to have a look at the other one?
What else we got around here? Oh yeah, actually this one does look like it has the pair of amphipods living on it, maybe. Yes, so living on the stalk of this worm over here, we've got a pair of amphipods that are probably a breeding pair. And one is they're currently in the middle of a domestic spat, and one is now moved off of the tube and is sitting on the seafloor. Yeah. One has been kicked out of the home, but they're taking some time apart. But but no, that's probably a breeding pair of amphipods. And they're often found attached to the sponges of stalks or tube worms or corals. And then there's a different type of amphipod close to the top here. And you can see this tube has much less. No, you can definitely see the hydroid end there. So yes, I think hydroid is the correct call on these. We can see there's a lot less hydroids on this tube. And then this one, the like feeding part has been pulled in, but you can still see what looks like it's operculum. I don't even know. Some kind of part of the tube. Oh no, maybe this is a sabellid. So this one has got the bristles that come out. Like, yeah. Cool. Yeah, let's see if we can see anything going on with this circle.
I feel like it's almost like a circular burrow that fits the cave in above.
It looks like, there's like a fish in shallow waters that does this. It's like
Yeah.
a puffer fish, I think.
Yeah, puffers will do it.
Weird. We've seen ring-like structures before, but not with burrows in them. Ring-like structures with, for instance, a black coral, because they're in one place, they consistently brush the seafloor around them, and that creates this ring-like, smoothened ring-like sort of area around them. But this is very different. This is literally holes in a circle, which is cool. Deep sea mystery. So I see there's a comment about not seeing any carnivorous sponges. We have seen some on yesterday's dive on the top of the Seamount. They were there, these thin carnivorous sponges, but we're not able to collect any, unfortunately. Another one of these sea pens, looks like a different brittle star living on it, but you can see here the two different ways of life for brittle stars, essentially. One is sediment dwelling and is a deposit feeder. So its mouth is on the underside, well buried. And it's able to just munch along the seafloor. And then the one on the left is a suspension feeder. And they hoist themselves off the seafloor, strain the water for food particles, and then bring them to their mouth using their arms. Cool. So that ring-like structure that we were looking at earlier, there's a term we use for that in science. It's Lebensspuren, which is basically German for life traces. And it basically is used collectively for any of these indications of life on the seafloor, whether it is poop or this group of circle of holes. But yeah, they are clues to what lives around here, even if we're not necessarily seeing the animal. How are we doing, heat flow team?
300 seconds to go.
What? Okay, good.
Time to look around.
Some more of these polychaetes. How good is your zoom? Could you reach the outcrop from here?
No, I could reach it, but it's going to be a little green.
What about that first rock? That was pretty good. That's great. So we can see on one of these encrusted lobate lavas, there's some animals, some polychaetes. These are different polychaetes, the ones we were just looking at. The ones we were just looking at, they make their tubes from the sediment, whereas the ones attached to these rocks actually make calcareous white tubes. Mm-hmm. Yeah, I feel like we haven't seen that many swimming things on this expedition so far. Only on Ascent and Descent. Like not so many like jellies or... You ready?
Oh yeah. That one's a little more spread out now.
We're at waypoint 5 right now. We're in the middle of a heat flow. Yeah, so that is a sabellid. And you can see it's got this beautiful plume of feathers that comes out. They're called feeding cirri, and they come out, I think, and they come out of the tube and essentially strain the water for food. Right, so we're at waypoint 5. We're doing a heat flow right now. We're trying to do a heat flow at every single waypoint, especially the ones where there's sediment, unlike at waypoint 1 and 2. And you smell nice. And then we are going to take push cores because this area, it does have elevated temperature. No, elevated temperature. The heat flow was hot. Yeah, so a lot of sediment's here. So we think it's a good spot. So we're definitely going to do push cores after this. And then Oda asked for one of the white sea cans.
Okay, so
And
the
then-
push goes where?
Right here.
In this
So
same
in
waypoint?
this, yeah, same exact. So probably won't even move
Okay.
much.
Okay.
And then we're right next to this little area, outcrop area with some hard substrate. And there are these tiny little white corals all throughout it. So we might pick up a rock with one of those. Yeah, see? With one of the tiny white corals on it.
So first the push cores and then the rock.
So first the push cores, then there's a white sea pen around that Oda wanted, some as well on the soft sediment. There's one like right close to us somewhere.
Okay.
And then there's some in that rocky area. There are all these tiny, tiny little white corals that look almost like lace. Yeah,
Okay.
see, that's one of the white sea pens down there.
Got it. Yeah.
So we could just grab that while we're sitting here.
Okay. Oh,
So you're going to be on the sampling. Watch. Lots of weird sea cucumbers we've never seen.
really?
Oh, okay. Yes. So yes. So we're gonna do after this. Let's do the 6 push cores, a Niskin.
6 or 8.
8.
Yeah.
However many is there?
Yeah,
8.
okay.
Then a Niskin. Then the sea pen. And then the coral.
Okay, let me write that down.
Should we do the distance first before we stir up?
Oh, sure.
Yeah, that would be
Yes.
good.
Sure.
So
Yes.
it will be.
We're gonna go into sampling. Niskin, 8 push cores.
Bienvenido, parcero.
Ah, parcero.
Rock for the coral. Rock for the coral.
We haven't done that. We haven't calculated.
Good morning, good afternoon, everyone. My name is Sergio Cambronero, and I will be joining you for the next hour. Thanks for staying tuned this live stream. We're live from 2,956 meters in a previously unexplored feature, a little bit southwest of the Dorado Outcrop. Thanks, Diva. Special thanks to my colleague, Dr. Diva Amon.
We're all done with it.
And all right.
We're gonna calculate the numbers so we know that it isn't.
And I would like to also give a special thanks to my colleague, Miguel Semedo, which is helping us with the annotations of the video. Niskin, yes, please. So we're currently making what we call heat flow measurements. We actually just finished. This is why you see what we call Excalibur buried in the ground. And this is also for us to test if the site is ideal for our heat probe analysis, which is basically a probe that sticks into the sediment and measures the heat transfer from the ocean crust.
Small, big, preference? One small and one big. 4 and 5.
Yeah. Yeah. One big. Yeah. One big as well. Yeah, got it. So currently we're gonna be doing a series of measurements of different parameters. So as I was saying, we just finished doing the heat flow measurement. Now we're taking water samples with the Niskin bottles. This is actually what you have on screen. The Niskin bottles are basically cylinders that we can trigger and close at particular depths. So we can have a little parcel of water that once we get back on board, we're able to do certain analysis. We will also be doing sediment sampling with the push cores, and then collecting some geological and biological samples.
4 and fire.
Okay.
Go to 3.
4 did it fire? Okay.
You want to do 3?
All right. Yeah, let's do it.
Code of fire, we just got no indication that it's fire.
Okay. What about 1?
You can either do 1 or 3, either one.
Yeah, yeah. Let's do 1.
Alright.
Let's just put here again. 5 did fire. Okay. So the 4 was that didn't fire.
And 1 fire.
Waypoint 5.
1 fired for sure. 5 fired for sure. And there's no way to tell them.
Hola, muy buenos días a todos y a todas quienes se conectan desde Costa Rica y otros lugares de habla hispana. Mi nombre es Sergio Cambronero, soy investigador y profesor en la Universidad Nacional y los voy a estar acompañando por la siguiente hora en este buceo, que es nuestro 7º buceo, si no me equivoco. Parte de esta expedición de Odisea de Pulpos. Actualmente estamos en una característica, por decirlo así, en un lugar muy particular, un poco al sur del área de estudio que teníamos inicialmente. Algunos le conocen como Caballito, eso es como nos aparecen en los mapas. Y básicamente estamos explorando este sitio que no ha sido explorado anteriormente. Así que, bueno, les agradecemos por la por la sintonía y por estar atentos a estos buceos exploratorios y al mismo tiempo invitarles que nos dejen sus comentarios, sus preguntas y por acá los vamos a estar leyendo. So what we have on screen are the push cores. So we did first the water sampling to avoid having any sediments in our bottles. And then we, because as you see, there's a lot of sediment that gets suspended after we take our push cores out. So this is usually takes about 20 minutes, so please bear with us as we see, oh, yeah, actually, can we have that coral first, just to not, because it's like gonna full of sediment. Yeah.
I can pick up and move a little bit. That way I can come up, get those push cores out, and get that in a better spot.
Okay, that's it. Yeah, yeah, that sounds good. Thanks. So we're gonna move a little bit with our ROV SuBastian, and special thanks to the Schmidt Ocean Institute here in the control room. We're gonna take this coral sample that you were previously seeing on screen. That's what we call a sea pen. And then we will take the push core samples just to avoid a lot of sediment on the coral. But yeah.
Yeah.
But yeah, sometimes it's pretty impossible to avoid. So bear with us. Also, it's a good time to tell you that all the sampling involved in these expeditions have gone through a strict permitting process of the Costa Rican authorities. So we also try to minimize our impact in the habitat and the marine biological populations down here. So pretty much all the samples that we're taking here are going to be deposited at the Zoology Museum of Universidad de Costa Rica, not only for us to be able to conduct our analysis, but also for other colleagues all around the world that might be interested in the samples to have the possibility of studying these organisms that we're collecting. This is our 7th dive of this expedition called Octopus Odyssey. So for those of you who have not seen our previous live streams, I invite you to go to the Schmidt Ocean Institute channels in YouTube and Facebook. You have all the recordings there. We have been seeing a lot of great observations. On the first dive, we were able to see a garden, an octopus garden with brooding eggs, and well, among many other things. So again, if you have not seen that and you're wondering how does that look like, everything is recorded, and you can access that completely free in the Schmidt Ocean Institute channels in YouTube and Facebook. So we are relocating the ROV just to be able to grab a better sample, as we see a sea cucumber that gets covered in the sediment cloud. Also, the lasers you see here are separated from each other by 10 centimeters. And this allows us to have an estimation of this of the size of things. And this is of great help, not only for taxonomic purposes to identify the species, but also to assess the biomass that we have at this particular depth. So please keep leaving your comments, your questions in our chats, and we will be very glad to answer them the best possible way.
Looks like we have quiver 2 next.
It is hotter.
All right, so we're gonna take a sample of a sea pen that we see here with an ophiuroid. This deep sea corals are usually associated with many hosts, as you can see here. And this also allows us to know more about these relationships deep down. At this particular place, we're currently at almost 3,000 meters.
Just to double check, quiver 2 is
Quiver 2? Yes, quiver 2.
okay. I'll make sure that
All right, thanks. Yes, I just didn't want to have like so much sediment around for example. Proceed with the push work. Yep.
That's good tip.
En su pantalla están viendo parte del proceso de muestreo de un coral de profundidad que se le llama pluma de mar. Hay muchos tipos de plumas de mar, pero particularmente estos son de aguas profundas y estamos interesados en identificar esta especie, particularmente la especialista a bordo, la doctora Odalisca Brady.
Pretty much the whole thing.
Can we have a closer zoom at it, if it's possible?
We did it earlier before.
Oh, really? Okay. Yeah.
We gotta get the zoom when that was all open.
All right. Perfect. Yeah.
You
Let's
wanna
go
go-
for the whole thing.
You
Yeah.
want us to go a little bit down into the mud?
Yeah, please.
Like so.
Yep.
Sometimes I have, okay.
Maybe a little too deep in the mud.
The whole thing.
Estamos viendo cómo colectamos una muestra de una pluma de mar y es un buen momento para recordarles que todos los muestreos como parte de esta expedición han pasado por procesos estrictos para obtener el permiso de las autoridades correspondientes en Costa Rica y al mismo tiempo comentarles de que todas las muestras son tratadas de la mejor manera posible para aprovecharlas, ya que muchos de estos lugares son remotos y, por ejemplo, esta es la primera vez que alguna persona ha estado acá con la capacidad de muestrear, así que tratamos de maximizar la integridad de estas muestras y muchísimas, diría que el 100% van a ser depositadas en el Museo de Zoología de la Universidad de Costa Rica. Así que no solamente es importante para nosotros tener información y poder hacer nuestros análisis posteriores, sino también para que otras personas del mundo puedan acceder a estas muestras biológicas si tuvieran el interés. El organismo que vemos asociado a la pluma de mar es un ofiuroideo, así que esto también es de nuestro interés, poder identificar estos animales que están asociados a los diferentes corales. Y estamos tomando esta muestra antes de proceder a tomar las muestras de sedimento. con los nucleadores. So beautiful sampling from our ROV team here in the control room. And bear with us as we will prepare to do next our sample, sediment sample with the push cores. Miguel, did you write down that it was with an opioid? With the host? Yeah. Yeah. Okay. Perfect. So what you see on the screen are our push core, and these are pretty much used to sample the sediments and to get a vertical resolution deep down the sediment layer. So if you take a closer look, these are cylinders that are inserted in the sediment layer, and in this way we're able to have a closer look at the different sediments that compose this layer. Sometimes they've been accumulating here for thousands and thousands of years, and this is of importance to know the history about this place, and also to make associations with the currents and the water column that we have up here. Also to remind you that we are 3,000 meters deep. And for us, it's not only important to assess what's down here at the bottom, but also, as I was saying, try to establish these connections between the water column and everything that's happening at the surface, because everything that happens at the surface eventually ends sinking. And this is part of what we call the biological carbon pump and the carbon flux in the oceans, which is a pretty recent research topic. So a pretty important objective also of this expedition. So we're going to go one by one, inserting this sediment push cores. So bear with us. Lo que ven en pantalla para los que nos sintonizan desde Costa Rica y otros lugares de habla hispana, estamos tomando muestras de sedimento con los nucleadores, que son estos cilindros que ven acá. Básicamente esto nos permite obtener una muestra de sedimento que nos permite ver cómo varía en la vertical a través de esta capa que se ha estado acumulando por años y años. En algunos casos, según nos han comentado nuestros colegas geólogos que están acá a bordo del barco, pues son procesos que se toman millones de años para poder tener estas capas de sedimento de este grosor, que pueden alcanzar entre 200 a 400 metros de profundidad. Y al mismo tiempo comentarles que gran parte de estos muestreos van a ser aprovechados por los colegas microbiólogos que tenemos a bordo, quienes van a estar también explorando y estudiando los microbios asociados a estos sedimentos. Microbios me refiero particularmente como parte de de estos microbios. Por ejemplo, tenemos bacterias, arqueas, también virus. eh, Ahora que los virus han sido un tema de interés común debido a la pandemia, eh, estos sedimentos en los fondos marinos alrededor del mundo se tiene evidencia de que son reservorios de virus, de los más grandes reservorios de virus en el mundo. Y también esto es de bastante interés, ya que nos interesa saber no solamente estos microbios que viven ahí, sino cómo los animales que se alimentan de estos sedimentos tienen la capacidad de resistir a todas estas bacterias y microorganismos que viven en los sedimentos. Em, y pues esto inclusive en un futuro no muy lejano podrían ser adaptaciones fisiológicas que se puedan investigar con métodos moleculares y que deriven eventualmente en posibles avances en medicina. Así que a nosotros nos gusta decir de que parte de la importancia de estudiar estas zonas profundas, no solamente desde un punto de vista ecológico y de conservación, sino que también con un alto potencial de bioprospección. Por favor, si nos sintonizan desde Costa Rica y otras partes, déjenos sus comentarios desde dónde se conectan, ya que también esto es importante para nosotros, saber el impacto que tienen estas transmisiones. Es la primera vez que esto sucede en Costa Rica, en esta particular área de los montes submarinos de la zona norte del Pacífico. Y esperamos que esto pueda divulgarse ampliamente, no solo en Costa Rica, sino en otras partes de Latinoamérica y el mundo.
It's funny how that makes a difference.
Yeah, you should be able to grab it at that same angle as well. Shoulder left some more. And then you have to, you don't want to see if you're in there. I mean, we'll lessen your weight.
So we're currently diving at almost 3,000 meters deep in an unnamed outcrop east of a previously identified bathymetric feature known as Caballito, which translates to little horse in English. And although this has been previously mapped, this is the first time that humans are able to come here and not only take samples, but also to see it live with this live stream. So it is an important milestone, not only in Costa Rica's history, but also for worldwide ocean exploration. And this is all thanks to the institutions involved, and in particular, the Schmidt Ocean Institute, who provides us with this platform for exploration and research called the Falker 2. And this submarine or ROV that is called SuBastian. This is our 7th dive of this expedition. And in this particular dive, we are aiming to investigate if there are signs of low temperature hydrothermal discharge using heat flow measurements, and with the secondary goal of seeing what kind of benthic fauna are living here. Y aunque no lo parezca, en este sedimento, en estas zonas, si bien no se logran apreciar muchísimos organismos viviendo en la superficie, muchísimos de ellos dejan marcas que pueden ser en la forma de agujeros, eh, rastros, huellas, inclusive sus heces, y todo esto es lo que nos indica también la actividad biológica asociada, ya que muchos de estos organismos a veces se entierran debajo de esta capa de sedimento. Entonces es importante, ya que a pesar de que no se vean, sabemos de que aquí hay una gran diversidad de animales y de comunidades biológicas que están viviendo asociados a estos sedimentos. Así que es para recordarles de que si bien no los estamos viendo, no quieren decir que, no, que esto no quiere decir que no están ahí. So we're almost finishing with our push course. We just have 2 left. So thank you everyone who's just tuning in and joining our live stream, live from the offshore, the Pacific coast of Costa Rica. This is our 7th dive of this expedition. So please, we will love to see your comments and questions in our Facebook and YouTube channels. And also, for us, it's really important to know from which parts of the world you're joining us, because part of the primary goals of these expeditions are not only to advance in science and research, but also to have a wider impact through these live streams. So we're just one left before we keep going with our sampling. After this sediment push course, we're gonna try to collect a rock sample or a geological sample. And this is also important for us to assess the minerals around this area that might tell us a little bit more about the history of this place. For example, how old are the rocks here, and if it has any association with outcrops and seamounts in the proximity that we have been exploring in this expedition. Y estamos insertando el último nucleador de sedimento. Y mientras nuestros colegas pilotos del ROV SuBastian terminan de insertar estos nucleadores, me gustaría hacerles una invitación a los que se conectan desde Costa Rica y otras partes de habla hispana que nos dejen en los comentarios de Facebook y YouTube desde donde se conectan y desde donde nos sintonizan. Asimismo, si tienen alguna pregunta, alguna duda, inclusive algún comentario, para nosotros es de muchísima importancia tener este insumo de parte de todos ustedes. Oh. So we see some interesting comments and questions in our YouTube channel that the accuracy of and precision of these samples and these sampling processes seem to be higher than in previous expeditions. This might be a question for the ROV team. If you have had any improvement
On what?
in the operating system of the manipulators, or if it's just that you've just gotten better because of practice.
Well, it's in. I'm gonna try to.
Yeah, try to push it on there. It was missing the collar.
Wow.
Yeah, you know that little black ring that binds them together?
Yeah.
Mm.
Kind of.
Yeah, you can try to put it in there. It'll seal if you can get that O-ring in there. It's not disrupted right now, and it's sitting on the bone, and you're in a good spot. I mean, yeah, it's been, we've had a lot of new guys, and everybody's just starting to get used to everything, and they're doing a fantastic job. And these have been some really trying shake-up cruises, you know, shakedown cruises, and lots of operations and lots of time. So, like, yeah, doing things like this normally you would freak out about, but you did a fantastic job.
Totally.
Just give it, just give it
So
a
we just got an answer from our one of our pilots here that definitely a lot of the improvement in the accuracy of this sampling operations is because of practice, and as they go and train more and more, they get better at manipulating and adjusting all the sensitivity and the controls. So special thanks to our team here in the control room, J-Rod and Scott, who are currently helping us here and controlling the ROV SuBastians. It is impressive the skills they have in order to make this possible. So again, special thanks to them and all the Schmidt Ocean Institute team. Y un gran saludo a los que se conectan, a todas y todos los que nos sintonizan desde Costa Rica. Pura vida. Y actualmente estamos en nuestro séptimo buceo de esta expedición, la llamada Odisea de Pulpos. Y estamos ahorita en vivo desde aproximadamente 3,000 metros de profundidad. Estamos explorando un sitio que que no había sido explorado anteriormente y que está asociado a un sitio que se conoce como Caballito. Esto es hacia el suroeste de donde nos encontramos anteriormente en los buceos que hemos hecho. Y si bien recuerdan, los que han tenido la oportunidad de ver los otros buceos, estuvimos inicialmente en el sitio principal de interés que se llama el afloramiento Dorado, en donde pudimos ver agregaciones masivas de pulpos incubando sus huevos. Así que si no han visto eso, les recomiendo 100% que se vayan a los canales de Schmidt Ocean Institute y puedan ver esas grabaciones. Lo bueno de todo esto es que si no lo logran ver en vivo, pues siempre quedan los videos ahí guardados. Entonces, ahorita estamos al suroeste de donde estuvimos inicialmente y esto es con la idea de tener también una mayor cobertura espacial y, y poder describir mejor esta zona donde estamos. Así que un gran saludo a los que se conectan de nuevo de Costa Rica. Muchísimas gracias por dejarnos sus comentarios. Esperemos que estén disfrutando estas transmisiones. También vemos que se conectan de otros sitios del mundo, como desde Barcelona, en España. Así que un gran saludo. Yo soy un gran, gran admirador del Barça. Así que me da muchísima alegría ver que hay gente desde allá. Y estamos finalizando nuestro muestreo de sedimentos. Seguidamente vamos a estar en este mismo sitio tomando unas muestras de rocas y luego vamos a seguir nuestro camino hacia la parte más alta de este sitio, que de nuevo está asociado a, a un lugar conocido como Caballito.
You enjoying your cruise so far?
Yeah, totally. It has been amazing. Every day is something new. It's just hard to work actually. You know, if you're like on your laptop trying to focus, just having this live stream is just
Yeah, when I was stuck in my room, I was trying to do some training, but watching the live stream as well.
yeah, man.
It's distracting.
Totally. Yeah, that Fauna for yesterday was mind blowing. Yeah, totally. I'm glad to have you back. You're feeling better?
Oh, I've been feeling better for a while.
Just yeah, taking precautions. Yeah. That's cool. That's cool. So we are currently retrieving our push core, and this is basically an instrument that we use to sample the sediment layer. After this, we're gonna proceed. Oh,
Yeah, it fell out as soon as it started coming out.
yeah.
You can clean it up.
All right. Okay. Makes sense. Thank you. After this sediment sampling, we will be aiming to grab a geological sample or a rock and then heading up to our next point. So stay tuned as we transit and explore this unnamed outcrop east of Caballito. And we might be seeing amazing creatures and seascapes. And if at some point you're not able to watch this live, just remember that all the recordings are available in the Schmidt Ocean Institute channels in YouTube and Facebook. There we go. So two main goals that our folks here on board the Falcor 2 do with this sediment samples are first to study and identify the microbial communities associated with these sediments, and secondly, identify and trying to see the organisms, microscopic organisms that we can find in the sediments. So this, let's say, remnant of organisms can tell us more about the previous conditions at this place, and even the ocean currents that have been present here, of that have had any influence on this area. So it is pretty interesting work of the geologists and microbiologists here on board. There is also my colleague Miguel Semedo from Portugal is conducting very interesting experiments also with these samples and trying to see if there is any particular effect on metals and metals exposure to the microbial communities that live that deep down here. So a special thanks to my colleagues Miguel Semedo, Rachel Lauer, and Rob Perring also that are joining me here in the control room. Y bueno, ya estamos a punto de terminar el muestreo de sedimentos. Nos faltan 4 nucleadores de recoger. Y después de esto vamos a estar tomando unas muestras de rocas y seguidamente seguiremos en nuestra exploración. So thank you everyone who's joining in our Facebook and YouTube live stream. The sediment samples that we're taking are definitely interesting because we have the possibility of studying the different, let's say, different stories associated with each layer. So, of course, as we can expect, the upper layer of the sediment is has been more recently deposited, and as we go deep in this layer, that sometimes can reach between 200 and 400 meters depth, we can see that there is a difference not only in the color, but also in the properties, the geochemical properties in this sediments. And also our colleagues on board take a closer look at these samples and for any evidence of organisms, small organisms, sometimes called radiolarians, that they leave their skeletons and in the past when they sank here, there is a record, worldwide record, to associate geological times where these microorganisms lived, and even ocean currents that brought them here to this place. So basically, the sediments are allows us to travel back in time and see what happened here during the last years, not years, but thousands and even sometimes millions of years. Sometimes this dates back to the Cenozoic, the Paleozoic. So yes, millions of years that we have the possibility with this sediment course. Millennia for sure. So we have some comments around the sediments here and YouTube channels. And yes, we're definitely, as I was saying, this is a window to the past to see everything that has happened here at this area. Let's remind ourselves that what happens and what has happened previously in the water column all over thousands and thousands of years, all the organic matter and the primary productivity in upper layers of the ocean, the majority ends up sinking down to the bottom of the ocean and building up this thick sediment layers. So all of this might be evidence, and this is what are we focusing on this cruise, evidence of different geological timescales. And as I was saying, if we are lucky enough, we might find some evidence of main ocean currents that are associated with this area, not only right now, currently, but also in the past. Y estamos terminando ya de recolectar las muestras de sedimentos. Nos faltan solamente 2 nucleadores. Y como estábamos comentando en inglés, voy a comentarlo un poco también en español. Estas muestras de sedimentos son de gran interés, ya que se hacen 2 análisis principalmente entre los expertos que están en este barco. En primera instancia, los análisis microbiológicos para entender qué tipo de microorganismos están asociados a las diferentes capas de sedimento. Y en segunda instancia, poder entender la edad y la composición de estos sedimentos. Recordemos de que estos sedimentos pues muchas veces están asociados a organismos y a materia orgánica que se ha ido hundiendo y hundiendo y acumulando durante millones y millones de años. Así que una vez que tomamos estas muestras, las traemos al laboratorio y se ven al microscopio, principalmente en busca de algunos organismos que tienen esqueletos o que tuvieron esqueletos calcáreos, como por ejemplo los radiolarios, bueno, que en este caso dejan exudados que se le llaman, o uses de sílice o de carbonato de calcio en el caso de los cocolitofóridos, y nos pueden decir más sobre la edad de estos sedimentos en las diferentes capas, pero también al mismo tiempo nos permiten entender las diferentes condiciones asociadas a la acumulación de estos sedimentos, por ejemplo, las corrientes marinas que las pudieron haber traído acá. So again, thanks everyone that's joining this live stream. we are very, very happy to see all your comments and questions. Also, we're happy to see where you're watching this from. And thanks again for joining us in this live streams as we explore the deep sea heritage of Costa Rica. We're currently in our 7th dive of this expedition and just finishing the sediment sampling. We will then collecting rock samples and then transiting to our next waypoint. So again, if you have any questions, comments, please leave it in our YouTube and Facebook channel chats. Entonces, estamos en el waypoint 5. De hecho, desde que llegué, aquí estamos atascados, por decirlo así. Entonces, tomamos muestras de Niski, solo para que sepa. Esta de Fire. Inicialmente la idea era hacer Niski, el Pushcore y luego el coral, pero como que el coral estaba muy cerca donde estaban los Pushcore, entonces decidí primero colectar el coral y luego los Pushcore. Entonces lo que hace falta, quién sabe, tal vez me dé tiempo a mí, quién sabe, de colectar una roca. Entonces están como unas que están como por ahí, están como más hacia el, como hacia el frente, están por aquí cerquita y son como, sí, como esos sí, bueno, sí, como esos lobby lavas que tenemos las pasadas, sí, que son así como rocas grandes. Colectar una de esas y ya luego seguir a ese juego. Y pues sí, creo que ya después de eso la prioridad es seguir haciendo los títulos, ya pues los sedimentos ya van a estar. Y sí. No, en cada punto están haciendo.
Por eso, cuando llegan a cada punto, reciben shimmering dependiendo de algún grado o algo así.
Ajá, sí. Y lo otro que voy a poner aquí, que si ve pepinos, que los colecte, porfa. Pero los que... ¿De cuáles? Sí, yo he hecho... Sí, no, de fijo, cualquier van en todo caso ahí me avisan. Sí, bueno, por favor. Pero sí voy a hacer eso, para mañana voy a imprimir las fotos para saber de cuáles han estado. Sí, ya. So we're finishing our sediment sampling. And soon we are going to grab a rock sample before we keep on our way exploring this unnamed outcrop east of Caballito. Yeah. And for those of you who wonder, Caballito stands or is translated to little horse in English. So pretty particular names. This is actually has been a topic here on board among the crew and the science party on how to name this unnamed features. And back in the days used to be much easier, but now has to go through a strict process with the global authority, which is GEPCO. So this will probably will take a while to see if there are proposals to name this unnamed features. But if this happens, the good thing is that we will try to have the input of many Costa Ricans in here for towards this naming. All right, so it is 10:55 in the morning, Costa Rica time, and for me, it was a pleasure to join you here in the control room in this live stream. My name is Sergio Cambronero, and thanks to my colleague Miguel Semedo from Portugal, who was joining me and helping us with the annotations. Soon I will be giving control of the dive to my colleague and friend of Universidad de Costa Rica, Celeste Sanchez, who will be joining you for the next hour. Así que muchísimas gracias a todos y a todas los que nos han estado sintonizando y viendo este buceo en vivo. Para mí ha sido un verdadero placer y honor acompañarlos durante esta última hora. Mi nombre es Sergio Cambronero. Soy investigador y profesor en la Universidad Nacional. Y especiales gracias a todo el equipo del Schmidt Ocean Institute y los colegas del equipo científico, especial al doctor Miguel Semedo de Portugal, quien me estuvo acompañando y ayudándonos con las anotaciones de los videos. Le voy a entregar el control a mi colega y amiga Celeste Sánchez, doctora Celeste Sánchez del CIMAR de la Universidad de Costa Rica, quien los va a estar acompañando durante la siguiente hora. Así que si tienen preguntas, comentarios, yo sé que Celeste está más que feliz en responderles. Así que pura vida.
All right, so we got a little bit of a handover happening here. Watch change. For those of you who are just tuning in, we are wrapping up some push core sediments. Push core sediment samples. About just shy of 3,000 meters below the surface of the Pacific Ocean off of Costa Rica. And we are currently at an unnamed outcrop. You're watching live footage from the remotely operated vehicle SuBastian, which is being operated by our pilots up here on research vessel Falcor 2. If you're watching along on YouTube or Facebook while we're live, please go ahead and type in the chat any questions you might have, and we'll do our best to get those answered. But right now, pilots are handing off as well, and it looks like our science handoff is just wrapping up.
Hi everybody, welcome to this next hour of the dive. I'm taking over now from my colleague Sergio Cambronero. My name is Celeste Sanchez Noguera. I'm a researcher and lecturer at the University of Costa Rica, and I will be here with you until noon local time. So at the moment, the pilots are going to be with us. It's our Adam and Taylor. And we want to thank J-Rod and Scott for the awesome work they have been doing over the past hours. Hola a todos, bienvenidos a esta hora de transmisión. Muchísimas gracias por seguir con nosotros. Mi nombre es Celeste Sánchez Noguera. Soy investigadora y profesora en la Universidad de Costa Rica y en este momento estamos en cambio de turno de pilotos. Entonces, básicamente lo que están haciendo es explicándose qué es lo que han hecho y qué es lo que tienen que continuar haciendo en lo que resta del buceo. Así que queremos agradecer a los pilotos salientes, J-Rod y Scott, y darle la bienvenida a Adam y Taylor, que van a estar con nosotros a partir de este momento. Para los que se acaban de conectar, lo que estamos haciendo justo en este momento es retirando el último nucleador de sedimento. Nosotros en cada sitio, dependiendo de si encontramos o no emanaciones de agua levemente más caliente, también aprovechamos para recolectar núcleos de sedimento. Estos núcleos son bastante importantes porque nos permiten complementar la caracterización química y térmica que estamos haciendo de estos sitios con una caracterización también a nivel de la composición de los sedimentos y de la fauna que se encuentra en estos sitios, así como también la microbiología de los sedimentos. Eso que están viendo ahorita en pantalla, de hecho, es, como pueden ver, una varilla, le dicen Excalibur, y es lo que se utiliza en estos momentos en el submarino para determinar si la profundidad del sedimento es la adecuada para poder realizar no solamente los núcleos, sino también las mediciones hidrotermales, que es uno de los objetivos en este buceo. So we are just retrieving the Excalibur from the sediment, and that's basically what the SuBastian pilots used to measure if the sediment in this area was deep enough, just to conduct or to do sediment push course, and also to measure heat flow. So we are going to continue now in our way to the next waypoint. Right now we are in waypoint 5, and we have still 5 more waypoints to go in this dive. So we just recently collected 8 push cords, and these are very important because they help us to characterize not only the composition of the sediments, but also all the fauna living in these areas. We also have colleagues who are doing some microbiological characterization of the sediments. And in addition to that, we are also sampling water with the Niskin bottles. They were just fired before the push course. And we are using that water to characterize the chemistry of these areas. En estos momentos están ubicando el submarino SuBastian en la posición correcta para continuar nuestro trayecto hacia el siguiente punto, que sería el punto número 6 de este transecto. Para el buceo de hoy tenemos una longitud aproximada de 900 metros. Son 300 metros menos de lo que teníamos en el buceo del día de ayer. Y el objetivo principal de hoy es hacer mediciones a lo largo de este transecto para poder determinar si efectivamente hay flujo térmico sucediendo en esta zona. Queremos recordarles que pueden también revisitar o rever todos los videos de las inmersiones anteriores en los canales del Schmidt Ocean Institute, ya sea en YouTube o también en Facebook. Y además invitarles a que lean los blogs que se están posteando en la página de ellos, en el sitio web. Son blogs bastante cortitos donde pueden enterarse un poco más de letras de hey guys, can we try to have a look at this? And okay, the lasers are on, so maybe Wendy, ¿podemos tomar un frame grab de eso con la escala, porfa, y después hacerle un acercamiento? So I think we're going to try to sample this. Yeah. This white coral that we have. Oh, I just broke it.
Oh, the big solitary coral.
This one here.
Yeah.
Yeah. So we have in the frame right now one of the octocorals that are one of the target species that we want to try to collect during this dive. So we are going to take pictures. The first one, the white picture with the scale. Remember the distance between the laser lights, it's 10 centimeters. And after taking the picture with the scale, we will proceed to taking some close-ups of the coral. Yeah, we have been seeing this coral in previous dives, but we weren't able to collect it before, and this is why we are going to try to collect it right now. Pero aquí hay rocas, están como bien rojas.
So we need to pick up the pace. Yep. So we need to deprioritize sampling so that we can keep moving.
Okay. So should we
I
start
know
this
that
way?
no. Okay. We should set the ship moving. We're behind in schedule and we need to catch up.
Okay. Okay. Good. But the rocks, I heard that you want some sample of rocks. No, nothing. Just keep going. Okay.
Those are not priorities.
Okay.
You can mark it if it's something really, really interesting to come back to.
Okay.
But we need to keep going.
Okay. Good. Okay. So we are not going to sample this. We are just going to try to keep going to the next waypoint. Okay. Okay. Thank you. Okay. So we are a little behind schedule, and that means that we are going to just continue moving to the next waypoint. We got already good shots of this coral, and we drop a marker in case we have to come back. Tenemos que movernos al siguiente punto porque, como les comentaba hace poco, la prioridad de este buceo es realizar las mediciones de flujo térmico y lo que hicimos fue tomar varias capturas de pantalla de este octocoral y poner un marcador de posicionamiento georreferenciado en caso que ocupemos regresar. Pero de momento entonces continuamos el camino hacia el punto número 6 de este transecto.
So in the chat, we have a question of how tall is the feature. Right now, let me pull up the map here. So currently we're at like 2950 for meters on depth. So you can see here, we're currently kind of working our way across this depressed area, little ski bowl here back in behind the peak. And so that's, you know, like let's see, what am I getting there? Yeah, like 2950, 2960 of depth. We're headed up to this peak. Well, we're headed up over here. And then if we have time, I think we might swing by here. That gets up to 2860.
I think we are already in waypoint 5.
So about 100 meters difference. But then the planes back here drop down to 3190. So there's kind of your relative depths of this area.
You're halfway now? No, this is 5 right here. We already did 5. We did 5.
But I think
we've been here forever.
Yeah, exactly. So we are now moving to.
Yeah, we're going to 6.
Yep. And that whole transect will end, terminate at an outcrop, which we're hoping to get to, which is part of the perfect target.
Hey, Alex, can you include again the map so I can give the same explanation in Spanish?
Oh, yes.
That would be awesome. Thank you.
Sorry.
No, it's okay.
There you are.
Ok, aquí pueden ver en el mapa inserto, eso es básicamente el transecto que estamos realizando el día de hoy. Cada uno de estos puntitos marca un punto o una posición dentro del transecto. Como pueden ver, en este momento nos encontramos justamente en el punto 5. Ahí está el pequeño avatar de SuBastian y nos dirigimos hacia el punto 6 para continuar en dirección hacia lo que sería el final del transecto que concluiría con el punto 10. Entonces estamos avanzando hacia lo que se ve como una cresta en el mapa. Y cuando llegamos a cada uno de estos puntos, lo primero que hacemos es ver con el Excalibur si hay profundidad suficiente en el sedimento para poder realizar las mediciones de flujo térmico, porque para poder realizarlas necesitamos al menos una profundidad de 1 metro. Thank you, Alex.
Yep.
So we want to take the chance to invite all of you who were not able to follow the previous dives to also visit the different channels of Schmidt Ocean Institute. Could be YouTube or Facebook. All the dives are recorded there. And we have also been posting blogs in the website of Schmidt Ocean Institute about the expedition. These are short blogs that were written by some of the scientists on board. So they will be available in both languages, in English and Spanish, in case you want to read more about the expedition. Como les comentaba antes, los blogs van a estar disponibles también en el sitio web de Schmidt Ocean Institute, tanto en inglés como en español. Son blogs cortos en los que pueden leer un poco más sobre la expedición. Estos blogs están escritos por varias de las personas científicas que están a bordo y de hecho también tenemos o vamos a tener un blog escrito por los 2 artistas a bordo. Para nosotros como costarricenses y latinoamericanos realmente es un orgullo poder ser partícipes de este tipo de expediciones donde tenemos la oportunidad de explorar y conocer un poco mejor lo que alberga el fondo de nuestro océano. Recordemos que nosotros ocupamos conocer para proteger, no podemos proteger lo que desconocemos y esto es parte de los objetivos que se tienen con este tipo de expediciones. El fondo oceánico en general es un ecosistema bastante desconocido, no solamente en esta parte del planeta, sino alrededor de todo el mundo. Y en los últimos años se han hecho varios esfuerzos por tratar de mejorar el nivel de conocimiento que hay de todos estos ecosistemas que hasta donde sabemos son ecosistemas bastante frágiles. So there is one sea urchin just right there, sitting on the bottom. Usually the soft bottoms, such as the one that we are just like seeing, are very good for spotting echinoderms, especially sea cucumbers. Sea cucumbers are animals that actually feed on the microbiomes and bacteria living on the sediment. So they just collect a lot of grains of sediment, they eat them, and then they ended up pooping cleaner sand. Los fondos blandos como estos que vemos en este momento en la pantalla son sitios bastante interesantes para observar equinodermos, específicamente pepinos de mar. Los pepinos de mar se alimentan de bacterias y microorganismos que viven en la arena. Entonces, como una estrategia alimenticia, lo que hacen es recolectar partículas de sedimento y una vez que han, digamos, adquirido todo lo que necesitan, entonces excretan arena que se encuentra, por así decirlo, más limpia. Entonces, muchas veces se dice que la gran mayoría del sedimento y la arena que se encuentra tanto en el fondo del mar como en las playas en algún momento han pasado por el tracto digestivo de un pepino de mar. Así que les invito a que presten atención y traten de contar cuántos pepinos pueden observar. En algunas tomas, de hecho, si prestan atención, se ven como unos chirulitos largos de arena, y eso precisamente son excretas de pepino de mar. Igual, de la misma forma, si en algún momento las ven cerca de la costa, es exactamente lo mismo. We are on our way to waypoint 6. We are actually just getting there. Today, the distance between the different points it's about 100 meters, half of the distance that we had yesterday in our dive. So once we get exactly on waypoint 6, we will check with Excalibur if we have enough depth just to conduct the heat flow measurement. Queremos aprovechar para saludar a todas las personas que están y han estado en sintonía a lo largo de estos días en los buceos. Sabemos que hay muchos hispanoparlantes que nos están siguiendo desde diferentes partes del mundo, no solamente desde Costa Rica, pero que también tenemos varios seguidores ticos que nos acompañan en este momento. Así que queremos darles un saludo muy especial, porque definitivamente para nosotros es muy importante aprovechar estas oportunidades que tenemos de abrirle el mundo del mar profundo, del océano profundo, a todas las personas que de alguna u otra forma también pueden contribuir a su protección. En este momento estamos a punto de introducir el Excalibur en el sedimento. Les recordamos que esta varilla la utilizamos para determinar si el sedimento en este sitio tiene la profundidad necesaria para poder realizar las mediciones de flujo térmico, que es uno de los principales objetivos de este buceo. So today dive it's going to be pretty much the same as the one from yesterday. We are working now in 12-hour shifts, which means that the dive starts at 5 local time, 5 in the morning, and it ends at 5 p.m. So we have to hit off bottom around 3:30 p.m. in local time. And our dive plan for today includes 10 waypoints. And we are now just like in waypoint 6.
So, are we good to go? We're just setting up the file.
Okay, awesome.
Okay, I'm recording.
Okay, so we are good to go. That means that we are going to be conducting heat flow measurements in this point.
So let us know when you're ready. We're ready.
All right, we're coming down.
Okay, so the heat flow probe is coming down. We are good to go. We got the green light from the pilots and also from the heat flow team that it's here with us in the control room.
Max probe. That's too low, isn't it? Not deep enough. No, we're missing the last sensor. So okay. Is that more if possible? I think that's full extension. Roll our pitch is 5. Pitch, yeah. Tell you what, let's pull up. I'll just swap it real quick. And I'll play with pitches and stuff to see if we can't do it. So what we'll do is we're gonna pull it up. I'm gonna pull up my arm. And then I'll just swap it real quick. We'll bump a little bit forward and we'll try.
Ok, al parecer les faltan unos pocos centímetros para poder tener la profundidad necesaria para hacer las mediciones de flujo térmico. Entonces lo que vamos a hacer es tratar de desplazarnos un poquito hacia adelante para ver si conseguimos la profundidad requerida. So we are missing a few centimeters apparently to be able to actually use or conduct the heat flow measurements. So we are going to move just a little bit forward to try to find the good spot to the heat flow probe. What is the distance between the marks? 10 centimeters? Nos confirman los pilotos que efectivamente el Excalibur tiene una longitud de 1 metro, que de hecho casi que lo mínimo que requerimos para poder realizar las mediciones con el sensor que mide flujo térmico. Vamos a ver si esta vez logramos la profundidad necesaria. El problema si no tenemos suficiente profundidad es que entonces el instrumento que se utiliza para realizar la medición de flujo térmico no puede quedar completamente sumergido y el instrumento es parecido como un tipo de varilla que tiene sensores en diferentes profundidades. Entonces ellos necesitan realmente medir en todas las diferentes profundidades para luego hacer una integración y poder establecer si hay flujo con diferentes temperaturas. Recordemos que nosotros lo que estamos tratando de identificar en estos puntos es cambio en la temperatura del agua que fluye básicamente por la corteza terrestre. El flujo de esta agua es de alguna forma lo que también permite que tengamos esta característica tan particular en el sitio conocido como afloramiento dorado, donde hay emanaciones de agua levemente más caliente. So the pilots are basically trying to find a good spot to conduct the heat flow measurements, and they're just saying, okay, that went a little bit deeper, so that's probably a good place. Yeah.
Yeah. Okay. Are you happy to right here? Yep. Yep. Okay, you ready?
Okay, so we're going to give it another try here.
Okay, we're coming down.
The heat flow probe is just right now going down. And yeah, that looks good. Yeah. Yay. Okay, that's perfect. So that means that we're going to spend about 15 minutes measuring in the sediment. Rachel, do you have to check there, or you want to come and explain what you are doing right now? Yeah. Okay, so I'm going to hand over the microphone to the expert in heat flow, who is going to tell you a little bit more about what they are doing today.
Can they see the probes on the screen? We can bring it up. Yeah. What is that? Okay. And the little guy?
It's just like a crab. I don't
A little crab? Okay.
I'm not a crab person.
All right, so this is Rachel Lauer. I'm here with Rob Perrin from the University of Calgary, and we are here conducting these heat flow measurements. This will take about 15 minutes, and these are important to understand the hydrogeology of the ocean crust, and in particular, how fluids are moving in and out of the crust and supporting the octopus and other sea life that we've been observing on these outcrops. So this area in general is about, or the chilled area that I'm going to describe is about 14,000 square kilometers. It encompasses 11 outcrops. And in this chilled area, most of the Earth's heat that is coming from the result of just the Earth cooling has been washed away through this process of hydrothermal circulation. And that hydrothermal circulation is water going in one outcrop, moving through the crust and coming out another outcrop or seamount. So this seamount we're on right now, we have measurements from 2001 and 2002 that suggest this area may be a discharge site. So where water is coming out of the seafloor, that water should be warm, because that water is going in one place where it's cold, picking up some of the Earth's heat as it travels and circulates through the crust and making its way to this discharge site where we are now, and it will be warmer than the background measurements. So if we put a bunch of these heat flow measurements together, we can start to understand some of the patterns that have developed between these sites of recharge and discharge. And it also gives us constraints or very specific temperatures that we can use to model these systems. So using math and equations and putting them together to basically create a picture of how fluids are circulating from the overlying ocean into the crust and then making their way out somewhere else. So yesterday, those who were watching, we were diving on Tango Zed, which is a very large outcrop. And based on heat flow measurements, it looks like that outcrop is both recharging, taking water in, and as well as discharging, which makes sense because it's a very large surface area exposed at the seafloor. And some of that water is not going to make its way all the way into the oceanic crust below. Some of it's going to go in, spend a little bit of time in Tango Zed, warm up a teeny bit, and then make its way to a seep, another place on the surface. And that's what we found. 2 places yesterday, water that was about 1 degree Celsius warmer than seawater. So that water had enough residence time in the seamount to warm up a little bit. I don't know what that was, but I'll let you step in if you want to describe the biology.
Thank you very much.
Yeah.
Thank you very much to Dr. Rachel Lauer for this incredible explanation about the heat flow measurements. Queremos agradecerle a la doctora Rachel Lauer por esta explicación que nos acaba de dar. Básicamente, lo que ella nos comentaba es que existen sitios donde usualmente hay entradas o tomas de agua levemente más fría en el fondo submarino y esta agua posteriormente es desplazada o fluye a lo largo de la corteza del fondo oceánico y en este trayecto básicamente el agua lo que hace es comenzar a adquirir o digamos transmitir más calor. Entonces, esta agua que ingresa con una temperatura en algunos casos de 2 grados en el punto de ingreso, posteriormente sale por otro sitio varios kilómetros después con temperaturas superiores, a veces de unos 10, 12 grados. Y esto es lo que nosotros hemos podido ver o lo que estamos también tratando de caracterizar en estos sitios. Se cree que el afloramiento rocoso del Dorado es uno de los sitios de salida y que por eso tenemos emanaciones de agua levemente más caliente y que sitios como el monte submarino Tengo Set que estuvimos explorando hace 2 días son precisamente sitios de recarga o ingreso de esta agua más fría. Entonces, para eso es que necesitamos hacer estas mediciones con este sensor que es bastante específico para poder determinar si existe o no flujo térmico en estos sitios. So what we had previously on the frame was it seems like a crinoid, but I'm not totally sure. And earlier we had something that could be either an hydrozone or a very small octocoral. So we have still few minutes to go here because the heat flow measurements take about 15 minutes, just as Rachel explained. And crinoids are also echinoderms, so they are related to sea cucumbers, sea urchins, sea stars, and usually they are attached to the substrate, but in some cases they are also able to swim away if they have to, and we have been able to actually see some of these crinoids swimming during the previous dives. Aprovechamos los 15 minutos que se necesitan para hacer las mediciones de heat flow para explorar un poco los alrededores de donde se está llevando a cabo la medición. Eso que estuvimos en pantalla hace unos minutos era un crinoideo. Los crinoideos son parientes o pertenecen al mismo grupo taxonómico de los pepinos de mar, los erizos de mar, las estrellas de mar. Usualmente los crinoideos se encuentran adheridos al sustrato, pero en ocasiones, si lo requieren, pueden desplazarse nadando. Y de hecho hemos tenido la oportunidad de ver en buceos previos varios crinoideos nadando. Esto que estamos viendo acá es un pepino de mar. Como les comentaba antes, en realidad es bastante usual ver pepinos de mar en fondos de sedimentos. Y tenemos un pequeño cangrejo que podría incluso estar alimentándose de los microbios o el microbioma que crecen en la parte exterior del pepino de mar. So we are just taking the time that the heat flow probe has to be in the sediment to look the surroundings of this area. This is, as you can tell, a sea cucumber. Let's see what else. Oh, can we take another close up of this polykid right here? Yeah, thank you. So we have like on the lower left corner, it's a polykid. That's a worm, and actually that's the tube that the polykid builds, and this is pretty interesting. Esto que estamos viendo en este momento es un poliqueto o gusano marino y ellos construyen un tubito, que es lo que ustedes pueden ver en este momento en pantalla, y lo que se ve en el extremo del tubo con un tono blancuzco, color celeste, es de hecho el poliqueto, parte del animal que construye este tubo. Entonces, el tubo está construido de diferentes materiales, pero muchas veces incorporan también parte de el sedimento y de otro tipo de partículas que se encuentren en el medio donde lo construyen. So we are just taking a close up of this polykit. ¿Le hizo un frame wrap? ¿Ocupa algo mejor? ¿No? Okay, let's see what else we can find in the meantime. Vamos a ver qué más podemos observar en esta área. Como pueden ver, si nosotros avanzamos rápidamente por el sedimento, pareciera a simple vista que no hay mucho que ver, pero cuando tenemos la oportunidad de detenernos y ver en más detalle, comenzamos a prestarle atención e identificar diferentes organismos que viven en este ecosistema. Además, los núcleos de sedimento que estamos recolectando también los vamos a utilizar para poder identificar la fauna que vive enterrada propiamente en el sedimento, que no se puede observar obviamente a simple vista, pero que también es bastante importante para caracterizar este tipo de sitios. Le recordamos que en estos momentos estamos trabajando con buceos en horarios de 12 horas, lo que significa que el submarino SuBastian ingresa al agua a las 5 de la mañana hora local y está de vuelta en cubierta a las 5 de la tarde también hora local. Entonces, las transmisiones comienzan aproximadamente a las 7 de la mañana y finalizan entre 3 y 3 y media de la tarde, cuando tenemos que comenzar el ascenso hacia la superficie. So how is it going?
100 seconds.
Well, I'm looking, do you have like the measurements like
Not so we're done. It looks a little bit colder, but
A little bit, not as warm.
Yeah,
we'll see.
We'll see in a minute.
Once we're done.
okay. Okay, so we are very close to finish the measurements with the probe, with the heat flow probe. Those white spots that we are actually seeing right now are also some sort of ooze calcareous sediments, and they are basically formed by a lot of different animals, like, for example, phytoplankton and foraminifera that were just like sink in the bottom of the ocean, and they basically form this kind of sediments or structures. We already got some samples of those formations, so the geologist on board has been looking at those under the microscope. And if I recall correct, the push course that we just sampled in the previous waypoint also had a lot of this white clay on the bottom. So going back to the crinoid, which is pretty nice to see actually. No, apparently the crab is just standing still. Okay, so we are done with the heat flow measurements. Now we can retrieve the probe from the sediment and continue our way to waypoint 6. Acabamos de terminar las mediciones de flujo térmico de heat flow, así que estamos retirando el instrumento que se utiliza para medir esto y procedemos ahora a retirar también el Excalibur y así podemos continuar el trayecto hasta el punto 7, que sería el siguiente punto en el transecto del plan de nuestro buceo del día de hoy. Cada vez de que nos acercamos o nos dirigimos hacia otro punto del transecto, los pilotos del submarino tienen que informarle al puente, que es básicamente donde pilotean la embarcación Falcor 2, porque entonces tanto el submarino como el barco tienen que ir desplazándose en conjunto. Entonces hay un trabajo de comunicación bastante cercano y constante entre el puente y el cuarto de control donde se encuentran los pilotos del submarino SuBastian. So we are now on our way to waypoint 7. Let's see if we can spot something interesting on our way to the next point. Remember, there is like a distance of 100 meters between each waypoint, at least for this dive that we are doing today. We can see some octocorals down there, a few brittle stars. Vemos algunos octocorales en el fondo, estos que salen como ramitas blancas. Algunos de hecho se puede apreciar que tienen estrellas quebradizas asociadas y también hemos visto algunas estrellas quebradizas que están directamente en el fondo, en el sedimento. Generalmente la diversidad de organismos que se pueden encontrar va a variar bastante dependiendo del tipo de sustrato. Entonces lo que encontramos, por ejemplo, en un fondo blando con sedimentos como este, va a ser diferente a lo que vamos a encontrar en un sustrato rocoso, como lo que hemos visto en buceos anteriores. Yeah, there are few sea pens just right there. Also more polychaete tubes. And sea cucumbers for sure. an hydromedusae apparently, just swimming in front of the camera. Oh, look at that. Varios de estos octocorales que vemos son, se les conoce como plumas de mar también. Ahí acabamos de ver en la imagen una estrella quebradiza moviéndose. Una de las maneras que ellos utilizan para desplazarse, ahí vemos otra, es mover sus brazos. Entonces parece como que si estuvieran caminando en el fondo submarino. Pura vida efectivamente es algo que se ha instaurado mucho en la cultura tica y es algo que nosotros utilizamos mucho en nuestro día a día. Acá estamos viendo un erizo, también un pepino de mar. Este pepino creemos que puede ser del género Oneirofanta, este que se ve como peludito. Con varias, digamos, extensiones. Vemos nuevamente en la imagen una estrella quebradiza desplazándose. So we just spotted a few echinoderms. Now there is another sea star. And just to comment on one of the things that people are just saying on the chat, definitely pura vida is something, an expression that is very installed in our Costa Rican culture. Actually, we use it a lot. It's not like a commercial brand or something that was just made up for touristic purposes. We actually use it a lot on a daily basis for a lot of different stuffs. So we are almost getting to waypoint 7, which means that now we are also going to try again with Excalibur and see if we can measure heat flow with the probe in this particular spot. Estamos en estos momentos llegando al punto 7. Así que vamos a, en unos pocos minutos probablemente, detenernos y probar nuevamente la profundidad del sedimento con el Excalibur. Acá en la imagen pueden ver un octocoral con varios equinodermos, varias estrellas quebradizas y también una estrella quebradiza de mayor tamaño cerca de la base del octocoral. Un pez que anda dando vueltas por ahí. Para darles una idea, en este momento el SuBastian se encuentra justo en el punto 7 y en el mapa podemos ver que la posición actual del barco es cercana al punto 6 que recién acabamos de pasar, lo cual significa que en estos momentos hay una distancia aproximada de 100 metros en términos, digamos, no de distancia profunda, sino más bien de distancia lineal entre el barco y el submarino. Así que procedemos nuevamente a medir con el Excalibur. En realidad es un nombre bastante apropiado. Recordemos que esto viene de la historia del rey Arturo y la espada Excalibur. Y vamos a ver si logra enterrarse un poco más. Pareciera que sí. Probablemente es un buen punto para tratar de realizar la siguiente medición. So far, one of the big predators that we have been able to spot here were, for example, the Pacific white scales. And actually, yesterday, we found one place that seems to be a reproduction site, or at least some kind of nursery. We found a lot of eggs from this species, and most of them were already open, but we were able to retrieve two of the eggs, closed eggs, and they seem to be, at least one of them seemed to be a viable egg. These deep sea species of skates, at least in other parts of the world, they are known to have periods of between 3 and 5 years of hatching, which means it's a really, really long period of time. And that's also one of the reasons why those species are very fragile. Okay, so the heat flow pro is just going down right now. And so we're still missing one of the sensors.
I can reposition.
So they are going to try to reposition the submarine again to get few more centimeters of sediment because otherwise the probe is not totally inserted into the sediment. Yeah, the eggs or the egg cases that we collected yesterday, there was one out of two seems to have a small embryo, or what appeared to be the beginning of an embryo. So what we did, we opened the cases, we documented everything with pictures, and then we also collected the small embryo and we placed it in a small vial with ethanol, and then we are going to run some genetic tests on the embryo as well. Estaba explicando que uno de los mayores depredadores o predadores tope que nos hemos encontrado en estos sitios son las rayas blancas del Pacífico. Y justamente ayer, al final del buceo que estuvimos haciendo, nos encontramos un sitio que pareciera ser un sitio de reproducción o guardería de estas especies, porque habían muchísimos huevos o restos de huevos de estas rayas. Las rayas, por lo menos esta especie en particular, lo que hace es que pone huevos y dentro de cada huevo se desarrolla un embrión que eventualmente va a eclosionar como una raya. Y entonces ayer lo que nos encontramos realmente fueron muchísimos de estos huevos ya abiertos, lo que significa que probablemente en algún momento eclosionaron. Y también tuvimos la oportunidad de recolectar 2 huevos que estaban cerrados. Cuando los huevos llegaron acá a la superficie, entonces los abrimos y en uno de ellos nos encontramos con la sorpresa de que en la yema se estaba comenzando a formar un embrión. Entonces, esto es un buen indicativo de que efectivamente eran o son huevos viables y que el sitio sí tiene las condiciones para que se lleve a cabo de manera exitosa el proceso de reproducción en especies o géneros de la raya como la que vimos ayer en otras partes del mundo, se sabe que los huevos pueden tardar entre 3 y 5 años para poder eclosionar. Entonces realmente esto los hace bastante vulnerables, también tomando en cuenta que son especies de larga maduración sexual, lo cual significa que igual tardan bastantes años en alcanzar su etapa reproductiva. Entonces, este tipo de descubrimientos que hemos hecho nos ayudan precisamente a determinar la importancia de estos sitios en proteger especies que son bastante frágiles. So, as I told you, the pilots are trying to relocate and find a good spot to conduct the heat flow measurements. Estamos en este momento tratando de encontrar un sitio adecuado para poder realizar las mediciones de flujo térmico. Parece ser que vamos a intentar en este punto medir la profundidad. Estamos en el punto 7 del transecto del día de hoy, el cual tiene un total de 10 puntos, lo cual significa que nos quedarían todavía 3 puntos más para finalizar este transecto. Y todavía nos quedan aproximadamente unas 3 horas de buceo. Lo que hacemos generalmente se hace un plan de buceo diario que se prepara con bastante antelación. Entonces, en el caso del buceo de hoy, que comenzaba a las 5 de la mañana, el plan de buceo ya estaba listo ayer al finalizar la tarde. Y básicamente el plan incluye los puntos para el transecto que se va a explorar durante el día. Además, se incluyen cuáles son las prioridades de muestreo. Así nos sirve de guía a todas las personas que estamos como líderes de buceo a lo largo del día. Y nos permite saber cuáles son las prioridades de colecta. Parece ser que no es lo suficientemente profundo, entonces vamos a tratar de continuar buscando un punto adecuado cerca de la marca del punto 7 y si no encontramos nada, entonces tenemos que movernos al punto 8. So apparently it's not good enough here, so they are going to try to continue looking around for another spot. And just here near the waypoint 7, and if we are not able to find it, then we will have to continue our way to waypoint 8. So we are just at the end of this hour of transmission. So I want to thank all of you who were here with us. Hopefully, you will be able to continue just watching this dive. And I'm going to give the spot behind the mic to my dear colleague and also friend, Dr. Jorge Cortez from the University of Costa Rica. So thank you very much to stay tuned with us. Estamos ya llegando a el final de esta hora de buceo, así que yo me despido. Quiero darles un agradecimiento por habernos acompañado y le doy la bienvenida a el doctor Jorge Cortés, quien va a estar ahora acompañándonos por la próxima hora. Acabamos también de hacer cambio de pilotos, entonces quedan en el cuarto de control nuevos pilotos, nuevo líder de buceo y continúa en la silla de tomadora de notas también la colega Gwendolyn Matamoros, quien es estudiante de maestría de la Universidad de Costa Rica. Muchísimas gracias y pura vida. Continúen siguiendo este buceo. Estamos en el 7, no hemos logrado encontrar suficiente profundidad para hacer el giro, entonces creo que vamos a seguir buscando un poco más y los que faltan son estos tres y la prioridad es seguir unos puntos para hacer el giro. Exacto, no hemos recolectado nada de momento.
Bueno, los núcleos sí.
Ah, sí, bueno, digo, de dichos. Exacto.
Good afternoon, good evening, good morning. This is Jorge Cortez from University of Costa Rica, taking over for the next hour. And we'll continue on our cruise here and looking for sediments, a thick layer of sediments to do some heat flow. And this is what we do is settle the ROV on the bottom. We find a thick enough layer of sediments and we lower this instrument that have very sensitive thermometers at 5 levels, and they measure how much heat is coming up from the ground, from underneath the sediments.
Looks a little bit, I think, yeah. I'll try to move on to another place. I mean, it all seems quite flat. Yeah. I'll try a different location. You can try this one. Like, are you able, I know that before you're kind of like propping it up with the arm a little bit. Does that work? Propping it up with the arm. Like on the skeleton? Is that what Adam was saying? No, I'm not sure. No, he was more like the gap. We were trying to find sort of like a feature where it's kind of like more of a mountain, so the probe's like just away from the surface, but as you can see here, it's quite hard and it's
Bueno, muy buenas tardes. Habla Jorge Cortés de la Universidad de Costa Rica, o buenas noches o buenos días, dependiendo de dónde están. Y voy a estar con ustedes durante la próxima hora, aquí narrando un poco lo que estamos haciendo. Y en estos momentos estamos cerca de un montículo sin nombre, a más de 200 kilómetros de la costa pacífica de Costa Rica. Y lo que estamos haciendo es buscando una capa de sedimentos suficientemente gruesa para poder hacer mediciones de transporte de calor. Aquí estamos viendo cómo esto que llaman Excalibur, como la espada del rey Arturo, se entierra para tener una idea del grosor de la capa de sedimentos. Y del lado lateral el ROV tenemos el medidor de temperatura. Y entonces es una varilla de metal que se entierra de titanio con 5 sensores muy finos de temperatura, y viendo las diferencias entre uno y otro se puede calcular cuánto calor está saliendo del fondo del mar aquí, atravesando estos sedimentos. Y esa es una de las misiones que tenemos en este viaje, hacer este tipo de mediciones y buscar sitios a donde sale agua más caliente, porque hemos visto que hay toda una fauna y flora, toda una fauna diferente, interesante alrededor de estos sitios. Entonces vamos a empezar el procedimiento. Bien. Ajá, ahí vemos en el recuadro, vemos donde está entrando el sensor.
I actually think it pushed us up a little bit there.
Yeah, I got all 5. Up it. I got
Okay.
a little bit of a response on 5. So.
Okay, yeah, we get a signal from all 5 sensors, so that's a good sign. And we're here at 2,955 meters depth. The water temperature is 1.83 degrees Celsius. And so this sensor is very sensitive. And they would just measure the difference at different heights in the sensor, in the sediments. Ahí pueden ver en el recuadro, ese es el sensor de temperatura, ya está enterrado en los sedimentos. Mide casi 1 metro y ya están los 5 sensores, se están registrando, y entonces ahora esperamos un rato para que se estabilicen y después se hace como un shock térmico para revisar que estén todo en orden y finalmente se hace la medición de cuánto calor está saliendo. Que estamos a 2.955 metros de profundidad y la temperatura es de 1.8 grados centígrados. Muchas gracias a todas las personas que están enviando saludos. Un gusto estar aquí. Thank you for all the people sending messages and greetings and questions. Keep that up, please.
Yeah, I'm stopping this one. I don't think it's enough of a response.
Okay.
So do you think we can pull it up and try and find a spot where it's
Yeah, we can keep going.
Okay.
The probe up now.
Yeah, the probe is coming up now.
It's definitely harder at the bottom there because it's sort of dropped the ROV as soon as we're.
Yeah, the measurement was not good enough, so we're gonna move on a little bit more to another place.
Thermistors.
Yeah, the temperature sensor, they're thermistors. Los sensores son termistores. Yeah, to measure how thick the sediments are, and in areas like this that we're not very sure, we use that rod that you saw at the end, that we call here Excalibur, and that goes in and gives us an idea how thick that sediment layer is. In other areas, there's data from previous expeditions and seismic data. So we know that some of these layers are several hundred meters thick. But in this area has never been studied before. That's why we have to do all these previous measurements. So here we're seeing some sea stars walking, octocoral, sea urchin over there. That's a squat lobster swimming across. And we see some rocks which are probably carbonates. The sediments in these deep areas, the top layer of sediments is very fine, and it's very easy to stir it up. Maybe people that have been following us have seen these look like sandstorms. Los sedimentos en las aguas profundas, las capas superficiales son muy finos y entonces es como muy fácil que se levanten cuando se desplaza el vehículo este. Y entonces parecen como tormentas de arena y tenemos que esperar a que se mueva esa nube para poder ver bien. Bueno, vamos a intentar de nuevo. I'm gonna try. That's Excalibur. That's the to see how thick the sediments are. So you see it going in. Mm-hmm. It's looking good. Ahí vimos el instrumento este llamado Excalibur, como la espada del rey Arturo, que es usada para medir usado para ver el espesor de los sedimentos, y ahorita vamos a bajar la sonda para ver el flujo de calor. Aquí con nosotros Rod y Rachel de la Universidad de Calgary en Canadá, que son los especialistas. Ahí podemos ver ya la sonda y va a empezar a bajar. We can see the probe as it goes down, buried into the sediment. Oh, yeah. Yeah. Yes, you can see the top part of the probe is it's very close to the sediments. That's what we're looking for. The heat probe is a titanium rod with 5 temperature sensors. El instrumento para medir estos flujos de calor es de titanio con 5 sensores muy sensibles.
I don't know what's different, but we were able to get 5. So it's still not, the fifth one's still not. Yeah, the fifth one's still not in.
Wow.
Well, I mean, we're gonna take measurement as it is, but it's something's, I don't know what the difference is between. And again, before it was like, it was barely making a measurement, it was frictionally heating.
Yeah.
And we don't, it's just, that last one is flat.
And this is a good inch or so
Yeah.
lower down than the very last one, because you could tell already that it was way too high, but this one looks better.
Yeah.
For sure.
Is that nut coming off of the Excalibur?
It looks like it maybe is, yeah. Loosened a little bit.
Yep. That diet, that's.
Get it right.
Yeah, we should try it.
That's a good point, yeah.
I've done it before, the bridge was with this. There's a single pump for it. Water temper. Drive up. Pull on. So the mower was then turned down. It's pretty much what you're doing. You plug it in, you turn it down.
I just like.
So I have to ball valve out, the handle, the pump handle is in handle pump valve. And they have that. Picked up the nut and then spun it back.
I remember to extend this big structure down subsea, we got all the way down to the bottom, like 2,000 meters or whatever. And they forgot to take the cap off the end. It was all bolted on, so they sent down a spanner to that fish tail on it. And we just had to get all the bolts off all the way down this thing. Huge fun, but took a while. Took a good while. It's hard to, we're getting on pretty well. Yeah. Yeah.
Some people here is asking if you can make a close-up of these sediments.
Yeah, sure. Now that it's cleared a little bit.
So you're doing a close-up of these sediments. Someone was asking. As you can see, they're very, very fine sediments. And fragments of this carbonate ooze. And in there, when we collect that and study it in details, we find many different organisms in there. Even if we don't see them, they're there. Plus, there is a lot of microorganisms. Here we see some little tubes from worms. It's like the more obvious thing that we look at. So some filaments, those are probably from worms. And here we're coming across tiene papeles, crustáceos. Yeah, yeah. That gives some rough idea of how it looks like. Yeah, the sediments here seems to be very, very fine.
Yeah. Looks to on around.
Mm-hmm. Mm-hmm. Yeah. I can see. Completely surrounded by sediments. Yeah. Thank you, Connor Johnson, that's been following us all these days. Suggests that what we were looking at before is it's a bryozoan covered with amphipods.
I'm surprised that the sensor is that looks really. That's the deepest I've seen.
The other ones were right on the edge. And it's normally we use it with that arm. Just put it over there. But it's already next to the bottom of the sub-bottom here. You don't want it sticking here. And it's steady. We need like two. Maybe one somewhere, but. Nice belly or something. But after 3 tries, use 4 out of 5. Same, keep going.
When we finish with this one, we just keep on moving to the next point.
Okay, we're happy. Finished.
Well,
Still
no,
going,
no.
or-
Let's still
Maybe
go.
in between them,
We're halfway done the measurement.
Okay.
Okay, cool.
but afterwards, maybe the blue spot instead of.
Yeah, here we're seeing a sea star that's partly buried. We only seeing the arms.
Not yet. Yeah.
Not yet.
We just fired the heat pulse. So.
Okay.
Yeah. was right between, yeah.
It's hard to say. Like I think it's usually the first two measurements.
Yeah.
Like, we'll see.
We're gonna just keep plugging towards the outcomes.
Yep. Yeah, we'll see if we can get more before we start going up.
Yeah,
It's maybe
exactly.
thinner again.
Yeah, I'm pretty sure it'll just peter out at some point, but
Hopefully it gets interesting.
yeah.
Yep. Yeah, right. This is pretty wild.
It was like the first two.
First two, about 3 times the sea level that we have in other places, so.
An easy way for me to like search versus think of like by time.
Yes.
I can also
Just like this Star Trek.
It does. Like Star Wars. If you don't know the scale, it could be the giant. Oh, there it is. Over there.
Oh, maybe that's where. Aha. Aha. Thank you. Now I know. And yeah. This is perfect.
We just walked with the.
I remember. Even value. This guy. Yeah. Yeah. Okay. But I have to take the time today, so I have to subscribe to the time.
I was in there a little amphipod going into that hole. And another one on the back. Estamos viendo un anfípodo bajando a ese hoyo, donde está parte de la estrella de mar. Pero en busca de comida.
We'll see-ish. It's more consistent with what we found at the other small outcrop, but the first two were.
Let's take a
They were further.
I don't know if we can zoom anymore on this.
Down.
Can you see it on here? Distance away that level. It's still there. Like just as much zoom.
Yeah, it looks like a brittle star. Coil up brittle star in the little cave on the right. Yeah, the scale is just a few centimeters. Let's see, we have it's clearing up, so we can see more now. You can see it's very small what we're looking at.
Ready to move to the next one?
All good, so we're ready to move. And we're gonna put up the temperature probe now. You could see there in the top left. As it's coming up, there it goes. Ya terminamos aquí, hacer esta medición y entonces está recogiendo el sensor de temperatura y el medidor de profundidad de sedimentos. Y ahora continuamos hacia otro de los puntos de estudio de la línea que tenemos planificada para hacer mediciones. Okay, we're ready here, so we keep moving towards another of our waypoints or another of the places where we have marked to stop and do some more measurements. In a few minutes, I'll ask Rachel or Rod to come up and tell us a little bit more details about these sort of measurements.
Okay. 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 I did an hour ago, but I can do it. Yeah. Yeah.
Oh, I see. Ah, sea star we haven't, I know, what is that? I know it's a tube anemone. Ah, that's beautiful. Yeah, and in a little while we'll have Rachel come over and explain a little bit about the heat flow and see if this is warmer or not. Ahora voy a pedido a Rachel de la Universidad de Calgary que se acerque aquí y nos cuente un poco más en detalle lo que son estas mediciones de flujo de calor. Y si efectivamente este lugar que nunca ha sido estudiado, pues se ven estos calores que suben. That was a polychaete you saw with something else hanging from it. So here we are at 2,957 meters depth and temperature There is 1.83 degrees Celsius, and it's pitch black. We can see what's there because we have lights on the ROV. But there's never light down here. Que lo que donde estamos ahora es a 2.958 metros de profundidad, una temperatura de 1.83 grados Celsius y en oscuridad total. Aquí nunca hay luz. Vemos más estrellas, este pepino de mar que hemos visto mucho, lo que parece una cola, pero es una extensión del cuerpo, plumas de mar, y vemos ahí un cangrejo corriendo de un lado a otro, y lo que se ven como unos rollos de sedimento, esos son los excrementos de pepinos. Here we see some sea pens, sea stars, and those rolls of sediments that we see once in a while. That's the excreta, that's the poop from sea urchins. Another one of those I think are anemones. Or checking on temperature errors. And a while ago they were talking about bioturbation. That's a process in which these sediments are stirred up by the organisms living there, the worms living inside the sediments, just pushing up deeper sediments to the surface, or the sea urchins just going through the surface, taking in sediments, passing it through the scot, and then leaving those trails that we see. The sea stars, as you saw, when they move, they stir the sediments. And so sediments are always being, where there's life, there's oxygen for these large animals to live. There will be, sediments will be bioturbated. Estaba explicando ahora un proceso que llamamos bioturbación. Eso es el proceso por el cual los animales que viven ahí están revolcando los sedimentos. Ahí vemos siempre hoyos en el fondo. Ahí viven diferentes especies de animales, principalmente gusanos, que están moviendo sedimentos de más profundo hacia la superficie. Pero también estas estrellas cuando se mueven distorsionan los sedimentos, igual los pepinos de mar cuando se alimentan toman sedimentos y después dejan estos rollos que vimos, que es un sedimento ya un poco diferente a lo que ingirieron, y los cangrejos que se mueven así, siempre están, mientras haya oxígeno y puede haber estos macroorganismos, va a haber bioturbación, va a haber alteración de esos sedimentos. That's a small jellyfish. Estamos viendo una medusa muy hermosa. Hemos visto muchísimas medusas en este viaje. Es una región muy productiva. Entonces mucho alimento relativamente mucho alimento en la columna de agua. We've been seeing a lot of jellyfish here. Last night, just before the end of the dive, we saw this very long, figured 20, 30 meter long siphonophore. It's a type of gelatinous plankton. A very, very long colony. It's a beautiful sea star there. 8 arms. One of those pits we've been seeing, some sort of excavation and exposing the calcareous ooze from underneath. And on the back we have a acorn worm. It's a hemichordate. Tenemos un hemicordado y eso que vemos al lado son las excretas del animal y ahí se puede ver el sedimento en el tracto digestivo. Yeah, we can see the poop from the animal. It's little sticks you see to the right, sediment sticks, and you can see sediments in the digestive system. So this one also are taking up sediments that have a lot of organic matter plus bacteria, and that's what they're feeding on. And sediment pass, they take the food out and comes out with a slightly different composition. There's a crinoid and some small octocorals.
Look around here right next to the place. I just see a little mound here.
Yeah.
Try and use this little star as a marker. Cool.
Yes.
Yeah.
Spotted like what looks like a good place to stop and do another one of the measurements. And here you can see a lot of those holes, the sediments, and there'll be either crustacean or worm down there, and moving those sediments up and down. Aquí vemos bien en el fondo todos esos hoyos que se ven. Esas son cámaras a donde viven ya sea crustáceos o gusanos. Que están moviendo los sedimentos y se puede ver ahí un trillo de algún otro organismo donde pasó y fue dejando ese trillo, una huella de una estrella, se ve muy bien. Entonces ven cómo van cambiando los sedimentos. Sí, una estrella interesante ahí con un brazo muchísimo más largo. Estas estrellas pueden regenerar los brazos, entonces cuando son atacadas sueltan el brazo, lo pierden y después lo pueden regenerar. Entonces uno ve esos tamaños diferentes de brazos y la coloración también es un poco diferente. Yeah, we were looking at that sea star with a very long arm.
I'm not sure. I'll have to wait for the best to figure out
The temperature of the sea star.
Yeah, like right at the last minute, I seen like an outline of a little star. He was in front of the mound, so I was trying to.
Yeah, here we can see the sea star I was mentioning. You can see a very long arm, shorter arms. They have a very high regeneration capacity, so when they're attacked, they will, in some cases, just let go part of the arm, or it's taken by the predator, but they're generated, so regenerated. So you could see different color and different sizes of arms. Okay, here we go with the Excalibur to see the consistency of these sediments. Mm-hmm. And by doing this, not only do we check on how thick the sediments are, but also help us stabilize the ROV on the bottom. Even at this depth of close to 3,000 meters, there are currents that will move the ROV a little bit. Con Excalibur, no solo medimos la profundidad del sedimento, pero se deja ahí para estabilizar el vehículo, que aún a estas profundidades, poco menos de 3,000 metros de profundidad, tenemos corrientes.
This looks like a mound, but I'm hoping it's not. A rock or something. It is slightly raised from the rest.
Right.
But
That's technically fine. As long as we didn't make the mound.
no, I was aiming for this mound. I kind of saw that sea star and was like, right, okay, so land near this. The sea star was like right next to this mound.
I guess we can watch and make sure if it is a rock, we can stop.
Yeah, if it pushes us up at all, just don't stop. Okay.
Ready? You ready?
Okay, I'm recording.
Okay.
Okay, coming down easy with the probe. Probe is on the move.
Definitely not a rock.
Great.
Hopefully, next time.
Looks good. Okay, so the heat probe going in. Now we have to wait for a little while for it to stabilize, start making some temperature measurement, and they give it a thermal shock. So the temperature goes up and then see how it decays. Meanwhile, we taking a look at this sea urchin with a lot of riders. You see a beetle star at the bottom. We see a barnacle attached to one of the arms and hydroids on the other side. So hitchhiking this sea urchin, more barnacles. If you look carefully at the end of the barnacles, there's these like fan structures. Those are their feet, and they use that to filter water.
Si en aguas poco profundas, uno lo que haciendo así, para poder agarrar, en cambio aquí se quedan, el corriente les va pegando. Hidroides y el sello acá.
Yeah, they. We can see it pulling out this structure, which are their feet, part of their feet, and we'll start filtering. I think there on the back we're seeing an anemone or a worm, not sure. You can see all these creatures just hanging on this sea urchin. Estamos viendo este erizo de mar con una gran cantidad de organismos que están viviendo encima. En el primer plano lo que vemos es el grupo de los cirripedios. Este es un percebe, que es como las bromas que vemos en las costas, en las rocas, solo que esto tiene un pie. Y eso que salió es la estructura filtradora, que son las patas del animal. Pues ahí al fondo parece como una pequeña anémona o un hidroide, no cuesta verlo. En el extremo derecho hay hidroides, es un grupo del filo de los nidarios, es el mismo grupo que los corales y octocorales, pero una clase diferente, hidrozoa, relacionados al coral de fuego. Y en la parte inferior del erizo vemos una estrella quebradiza. O eso que yo decía que parecía una pequeña anémona, más bien debe ser un crustáceo. Le estoy viendo como unas patas. Aquí vemos el cirripedio con las estructuras afuera para alimentarse. Okay, if you look at the blue star, there's like a white blob there. That's probably an amphipod. Okay. Changing pilot.
Yes, we just went to the midpoint because by the time we got up place last time it was around about the midpoint. That's why we're not actually on waypoint 8. So this, try to land on a little mound there. Most of the bits of seabed are like too far away. The photographs we've been in coverage from the last program.
All right, we've got a little bit of a watch change happening here. ROV pilots and scientists both conferring with their predecessors as they take their places in the control room here. For anyone just tuning in, we're about 3,000 meters down on an unnamed outcrop. And you're watching live footage from remotely operated vehicle SuBastian being operated from research vessel Falcor 2. We're just making our way across a little bit of a bowl or depression on the top of this outcrop. We're halfway between waypoint 8 and 9 out of 10. In fact, I can put the map up for a moment here. All right, ROV update location there. You can see where we are there. We're going to start heading up the slope here shortly. And you can see that we've made our way across this little depression here at the top of this outcrop. For everyone in the chat, thank you for joining us today. We do love having folks explore along with us. There's been some fun questions in the chats. So we do stream on YouTube and Facebook. And if you're ever watching live, feel free to type questions in those chats and we'll do our best to get those answered for you. Scientists are still swapping over there, but the ROV pilots have finished up. Well, it looks like Jorge might still be in the science seat for a little bit. And then we've got Chris on pilot, and Carl on co-pilot, and as data logger, my name's Alex.
Well, it's the end of my shift. I'm Jorge Cortez from the University of Costa Rica. Thank you for being tuned with us here. And now it's
trying to focus past the sediment here.
University of Costa Rica will be taking over. And with her, it will be Nadia from Brazil, making notes, and with the temperature probe, Rod and Rachel. Bueno, muchas gracias por haber estado con nosotros. Les habló Jorge Cortés de la Universidad de Costa Rica y ahora le doy el campo a María Isabel Sandoval, profesora de la Escuela Centroamericana de Geología de la Universidad de Costa Rica, y la va a acompañar Nadia de Brasil, y para las mediciones de flujo de calor van a continuar Rod y Rachel. Muchas gracias y nos oímos mañana.
Sí, ya me anunció. Ah, ok. Sí, sí, sí.
Hola, muy buenas tardes. Les habla María Sandoval de la Universidad de Costa Rica. Continuamos en este camino hacia la cima o parte más altas de un nuevo montículo submarino sin nombre. So, hello everyone, this is Maria Sandoval from University of Costa Rica. Here with me is also Mayla Guillon. She's a biologist from the Research Institute for Sustainability from Potsdam in Germany. And so we are in our path, in our way to the top on the summit of the unnamed small mount here, submarine seamount. So to tell you something about the sediments here that we are looking at are especially some oozes from probably diatom, foraminifera, some calcareous nanofossils. The part that is whitish is very rich in nanofossils and foraminifera, and there is full of echinoderms here, especially brittle stars and some urchins. Para contarles un poquito de lo del sedimento que estamos viendo por acá, estos sedimentos son principalmente acumulaciones de organismos pelágicos, de lluvia pelágica, por ejemplo, diatomeas, foraminíferos, planctónicos, algunas, puede haber radiolarios también, y cocolitos. En la parte blanca que han estado viendo en el camino que hemos estado recorriendo, es principalmente un barro calcáreo rico en nanofósiles y en foraminíferos. Aquí lo que estamos viendo es una ofiura y hemos visto varias, sobre todo bastantes equinodermos en este sector. Okay, cool. So here we are waiting for the heat flow measurements. So we can continue looking at these beautiful brittle stars. So the ophiurids here, they live there and in this little hole. So they are filter feeders and they are nourishing here. Aquí, bueno, vemos una estrella quebradiza o como le llamamos ophiuroideas. Y está viviendo, este es su lugar de vivienda y aquí estaría en una posición de alimentación. Recuerdo que son organismos filtradores. Okay. So we had already the heat flow measurement. So now we will continue with the next point. And hopefully we will start collecting samples too. Yes. Okay. Yeah. Bueno, con respecto a las medidas de temperatura que hemos estado teniendo en este sector o medidas de flujo térmico, hemos en este sitio lo que se ha observado es un incremento en esta medida, por lo tanto se puede indicar que o es un indicativo de tener algún tipo de descarga de agua un poco más caliente que lo que hemos tenido, por ejemplo, en el sitio de ayer, que era Tengo Sed. Aquí vamos a durar un poquito haciendo ajustando la parte superior del Excalibur porque está un poco suelta. Así que podemos observar esta maniobra muy interesante que van a hacer los pilotos con su excelente habilidad. También les comento que acá está mi compañera, la doctora Mayla Guillón, que es del Instituto de Investigación de Sostenibilidad en Potsdam, Alemania. Y Mayla está observando y anotando todas las especies, tomando el muestreo y demás, mientras que yo les voy contando un poco de lo que vamos observando en el fondo oceánico y tomando las muestras. También mientras aprovecho para contarles que sigan que sigan las redes y también los blogs que hemos estado escribiendo, parte de la parte científica de investigadores. Ya han salido varios blogs de esta expedición Octopus Odyssey y los invito entonces a leerlos. Están en ambos idiomas, en inglés y en español.
So the
I
pilots
wanted to
here.
invite you to read some of the blogs that we have written in all this part of these days. So you can take a look in the Schmidt Ocean page. And there is also some blogs the blogs are written in Spanish and in English. So here they are trying to adjust the Excalibur to continue with the measurements. So we will take a little bit more time here.
I mean, that's all right. Yeah.
Hoy tenemos el propósito de recorrer 10 puntos Estos son separaciones de 100
Oh,
metros
bottom one stopped spinning.
y ya vamos camino hacia el 9º punto, por lo tanto ya iniciaremos un poco con lo que es colecta de muestras en estos últimos 200 metros que quedamos de recorrido y esperamos también medir un poco más de hacer mediciones de calor térmico, de flujo térmico. So here for today we have 10 points. Oh, great. Our purpose today is to visit 10 waypoints. So now we are going to the 9, and in 200 meters we are almost done. So we will we will be collecting some samples and also doing some heat measurements, heat flow measurements. Saludos a todos los familiares, amigos y amigas que nos escuchan. Tenemos bastantes personas conectadas de Costa Rica y de otros sitios en Latinoamérica y el mundo. Entonces agradezco para darles un saludo. Por ahí está mi mamá. Un abrazo. Nos escuchan bastantes personas también de otros sitios en Latinoamérica hispanohablantes, por lo tanto aprovechamos estos momentos en que tenemos a bordo bastantes investigadores nacionales, como también tenemos otros investigadores internacionales, pero aprovechamos que somos hispanohablantes para enriquecer lo que es y dar a conocer esta visita al fondo oceánico en las afueras de Costa Rica. Estamos a 200 kilómetros aproximadamente fuera de costa de Nicoya, de la península. Y en este sector, como les recuerdo, tenemos algunos volcancitos o extintos, ¿verdad? Algunos montes submarinos y montículos submarinos que hemos podido estar visitando en estos días. Ha sido pues maravilloso todo lo que hemos podido transmitir hacia ustedes y nosotros también conocer por medio a altas profundidades. En este momento estamos a una profundidad de casi 3.000 metros, 2.956, que pocas veces podemos visitar estos sitios o ver estas hermosas imágenes. Así que los invito y las invito a quedarse conectados. Y continuar viendo estas increíbles imágenes y organismos. Okay.
The first 3 we had to skip because there was only 20 centimeters of stuff in it.
Les cuento también que aparte de tener un equipo interdisciplinario, como nos saludan nos dice una de nuestras espectadoras, también tenemos 2 artistas a bordo que se inspiran en el fondo oceánico en estas imágenes. Pueden también seguir sus trabajos y parte de sus blogs, porque van a tener blogs escritos. Estos artistas son impresionantes, estas pinturas también. Y esa inspiración que les ha traído conocer todos estos fondos y todas estas animales tan interesantes.
So it's starting to look a little rockier here. You want to continue on, or
how far are we from the last one?
Un abrazo ahí a mi familia que me está saludando. Besos a todas, abrazos. Claro que se pueden socializar, se pueden seguir las páginas de los artistas. Uno de los artistas es costarricense argentino, y se llama Carlos Hiller. Y tenemos también, ya les digo el nombre exacto porque solo sé que se llama Michael, pero es estadounidense. Y ya les indico el apellido para que puedan seguir sus páginas y sus redes sociales también. Michael Droch. Michelle, así, Michelle Droch. Yes. Y Michelle trabaja como profesor o lecturer en arte y en cultura de arte y visualización en Estados Unidos, en el Bates College. Aquí estamos viendo un pulpo por acá. So we are looking at the first octopus. So can we okay, so estamos viendo el primer pulpo que vemos en este sector. Lo vemos ahí caminando en el fondo oceánico y cercano a las rocas. Por lo tanto, aquí iremos con bastante cuidado. Recuerden que esta expedición, el principal objetivo es justamente ver esta asociación que tenemos, los que tienen estos organismos con estos fuentes de aguas termales, podríamos decir, o un poquito más calientes de las aguas circundantes. Okay. Aquí tenemos a la par, ya llegó la experta de pulpos, Janet Voigt, que nos está indicando y va a estar acá posiblemente con nosotras para continuar observando y siguiendo los movimientos de los pulpos. También, si ven ustedes más pulpos ahora que seguimos el camino, nos pueden indicar. So here we have Janet in the control room. Janet Voigt, she's the specialist. She's one of the biggest specialists in octopuses in the world. So she will be around and trying to see if we can see more octopus, and also if we can, maybe we can collect one depending of the position and what they are doing.
You get a nice picture and move on. Yep. Alright, nice picture and then we'll move on. Well, we'll not move on. We'll we in a spot. Pretty. They all are. It's like
It looks a little bit different, right?
Do we like do a heat flow measurement right here? Yeah, that's the one. Yeah.
It's just like the top of sit. Gotcha. Probably put in a little bit more and do that. We'll get rid of all that sediment.
Bueno, excelentes imágenes acá de este pulpo. Está moviendo, se está retrocediendo un poco con los brazos.
So we don't need to spend a ton of time getting rid of this. We have other things we can do. Yeah.
Okay, I will continue.
Yeah.
Yeah.
Especially if it's so bright.
Es hermoso. It's super cute. Yes, I agree. So for now, he's in a, we can say, in a quiet position, just resting.
Yeah, we're waiting to heat that there.
It's weird. It's really weird.
That's so-
That's why it doesn't work sometimes for you guys.
I did that before and it switched every time I turned it, it turned the auto back on. I don't know why.
That's why it gets back. So you're back.
Alright, so we'll spin.
Well, we're gonna do one more heat flow here and then, yeah, we're gonna try it.
All right, so we're hoping to get another heat flow probe done here before we move into these rocks. Obviously, deploying the heat probe down into the rocks wouldn't work too well, or the heat flow probe. Yeah.
It sounded like.
We will try to measure here another heat flow measurement. So we are trying to get a good spot to try the thickness of the sediment. Bye bye, octopus. Yes. See you later.
You know what, if you're diving, I think this is.
Yes. To answer the question of Connor Johnson, has the heat flow been warmer here? So yes, for the moment we were having warmer heat flow measurements. So probably, the idea is to get some more measurements. So that indicates that is a outflow spot here in this little seamount. It's not as higher as Dorado, but is warmer than Tango Set, for example. Aquí estamos observando un poquito, ya vieron cómo cambia el sedimento del sedimento completamente. Ya venimos aquí con un tipo lavas basálticas nodulares, y ya podíamos ver cerca donde estaba el pulpo algunas rocas ya más masivas basálticas. Les recuerdo que estos son rocas que se originan en la dorsal medioceánica, donde se genera corteza, y esta corteza es de tipo basáltica, tiene poco sílice generalmente, y después, bueno, estas forman como estos montículos a veces, también grandes cantidades de corteza, ¿verdad? Y estos montículos o montes submarinos es lo que estamos apreciando acá, con cierto relieve comparado a la corteza que es más plana, digamos. Vamos a tener esa parte plana, pero también algunos montículos. Y en estos montículos es donde hay ese ingreso de agua en algunos sectores, y también hay como una salida de esa agua fría en sectores como el Dorado, que es un sitio tan especial y donde fue que apreciamos muchísimos pulpos también. Entonces, esto es un poco la conexión que hay entre estos montículos, y por eso es que los estamos visitando, para poder entender cómo funciona este flujo digamos, este flujo a través de la corteza. Aquí estamos tomando otra medida de flujo de calor, así que vamos a esperar 15 minutos. Así que por mientras los dejo apreciar algunas imágenes del fondo oceánico, ver algunos organismos que nadan, que están ahí simplemente descansando, como las estrellas quebradizas o los ofiuroideos. Sí, aprovecho para comentarles, ahora que tengo el comentario de mi hermana, un saludo a mi hermana también, que es maestra. Acá en el barco hemos tenido bastantes programas de lo que llaman Ship to Shore, que es entonces tener o que las estudiantes de escuelas o inclusive de colegios han tenido la oportunidad, tanto de Costa Rica como de Estados Unidos y otras partes, han tenido la oportunidad de conocer el barco o hacer un tour del barco desde acá. Se hace de manera obviamente remota, y se les muestra los diferentes lugares y sitios del barco. Por lo tanto, les dejo la posibilidad de solicitarlo tal vez más a futuro para la expedición de diciembre. Les recuerdo que estaremos regresando en septiembre 2 semanas, y lo que es ahora estamos 3 semanas. Ya llevamos, lo que fue, zarpamos del 2 de junio y ya llevamos unos 10 días acá en el barco. Bueno, aquí observamos un pequeño octocoral y una esponja a la par. Ahí mismo estamos observando lo que es una ofiuroidea debajo o pues muy cercana a lo que es el octocoral. Un pequeñito, ahí vemos un pequeñito, olvidé el nombre de estos, sí, un camaroncito. Ujú. Sí, las cámaras que tiene el robot son muy alta resolución, por lo tanto podemos ver detalles, bastante detalle de los organismos. Este organismo podría medir tal vez, no sé, 3 centímetros, la verdad no sé, pues tendríamos que ponerle láser. El robot también cuenta con un láser que se activa. Son 2 puntitos verdes y esos 2 puntitos verdes miden 10 centímetros. Entonces lo que hacemos cuando vemos un organismo que nos interesa es principalmente tomarle la foto con los láseres y saber el tamaño. Como lo vimos antes con el pulpo. And if we take a look of these sediments and that we are collecting to, in the sediments are many other creatures, organisms that are also amazing to study and that can say many things, like for example, foraminifera, radiolaria, and some part of the phytoplankton and also some protists. So part of our studies here is to study those sediments and to try to identify what we have here and those sediments and the microorganisms, the physical property, if it's more sandy, or if it's the grain size also, and the minerals, the mineral content too. So en estos sedimentos que hemos colectado, identificamos, por ejemplo, microorganismos, también identificamos minerales, las propiedades físicas, como por ejemplo el tamaño de grano, si fuera más limoso, arenoso, y todo esto lo hacemos desde la Escuela de Geología de la Universidad de Costa Rica. Tenemos un equipo que trabaja de varios investigadores de la escuela, que trabajamos con esta parte también asociada a este gran proyecto de este sector. Estamos a 200 kilómetros de la costa de la península de Nicoya, mar adentro. Les comentaba ayer que estas son algunos lavas de tipo nodular basálticos y no como se ha creído que son, por ejemplo, nódulos de manganeso. Entonces, nada más para aclararles y comentarles esta curiosidad. Do you want to explain something in English about the heat flow? Or there are some people asking for the heat flows, getting warmer or something.
Oh, yeah. Sure.
Okay.
I feel like everyone's bored of me saying the same thing.
I will never be bored of you saying this.
Oh,
Yeah.
that's
Okay.
all right. I'll talk.
Okay. Los dejo con la doctora Rachel Lauer, que es la especialista en estas medidas de flujo térmico. Va a hablar en inglés. Después les cuento un poco lo que dijo.
Yeah, in English.
Yes.
My Spanish is horrible. Hola, this is Rachel Lauer. I'm here with Rob Perrin. We are responsible for conducting these heat flow measurements on this newly explored outcrop. And what we found so far on this dive, again, these are preliminary results, are that heat flow has started fairly high at higher elevations, dipped down a little bit, and is now increasing again as we come closer to the other side of this sediment pond that we're cutting across. And why this is important, the heat flow actually gives us some clue as to the temperature of the outcrop itself. And when we see temperatures below what we would expect, we can guess or be more confident that this particular location has cold bottom water going into the outcrop or recharging. And in the case of this outcrop, we think that warmer water that's moved through the crust, picked up some heat, is actually coming out or discharging through this unnamed outcrop. We have done heat flow in 2001 and 2002. Well, not the royal we, not me personally, around this field of outcrops. And only a couple of measurements were made on this particular site. At the time, those were relatively low compared to the measurements that we have today. And these measurements give us a little more confidence that this site is a place where fluids are coming out. And that is important in this particular study because another outcrop that we visited the first 2 days, Dorado Outcrop, is the site of these octopus gardens, where brooding octopus are lining the cracks in the rock with their eggs and lying in the brooding position. And I don't think we have an estimate of how many octopus were located on that particular outcrop, but I think it's safe to say well over 100. So this is what started our interest in looking for additional sites where warm fluid might be coming out. Oh, is that shimmering? No? No. So we're zooming in particular, looking for cracks where we might see shimmering as a result of that warmer water that's heated up in the crust and is coming out of the outcrop. When it hits the cold seawater, even at temperatures just above seawater temperature, you can see some shimmering. And that tells us that these little springs or seeps are locations where that warmer water is coming out. If we do find them, we would like to zoom in and potentially take a sample. So we'll continue
after this heat flow measurement, heading towards higher elevation on the outcrop and looking for more potentially sites like we encountered at Tango Said, where we found 2 new springs on that outcrop, where the water was about 1 or 2 degrees warmer than bottom water temperature. Anything else? Yeah, for those who are just joining and haven't heard me say this maybe 20 times, the heat flow measurements occur in 2 parts. We actually take the lance, the 60 centimeter lance, and insert it into the sediments. The result of that is a frictional heating. So we get an increase in temperature, and along that lance are 6 thermistors, or high precision temperature measurement devices. Wow, that guy's really cool. And those measure the frictional heating, and at about 7 minutes after that frictional heating, we can estimate the thermal gradient. So how temperature is increasing below the sediment. Then we fire a heat pulse at 7 minutes, and we know exactly how much energy is going into that heat pulse. And then we measure the thermistor's response to that pulse of energy, and the way that that decay happens tells us, or allows us to calculate, the thermal conductivity. And the product, or by multiplying thermal conductivity times the temperature gradient, we get heat flow. All right, I'll let someone come back and talk about the fish and other things that are happening that are probably more interesting to most of our listeners, but good to chat with y'all again.
Bueno, muchas gracias a la doctora Rachel Lauer, que nos habló y nos explicó súper detallado todo lo que estamos estudiando del flujo de calor en estos sectores. Como ya les he explicado, buscamos principalmente sectores donde el agua sea un poco distorsionada o brumosa, y por ahí entonces podría haber una salida de calor, como lo vimos por ejemplo en el sitio del Dorado, donde había esta cantidad de pulpos anidando. Ahí entonces teníamos una salida de agua caliente que ellos aprovechan para poder entonces cuidar a sus huevos y poder tener sus pulpitos. Esta es la primera vez que se ve y que se observa este tipo digamos, de pulpitos saliendo y viviendo en el sector. Así que para nosotros ha sido un descubrimiento súper novedoso. Y ok. Además, tengo una pregunta en el chat sobre el tamaño de estos pulpos. Bueno, este pulpo que vimos hace poco, bastante grande, puede medir tal vez hasta 1 metro, o sí, unos 80 hasta 1 metro. Y por el momento hemos visto al menos 2 especies de pulpos, ¿verdad? Estos pulpos con ojos más grandes y como más, pues diría yo, abultaditos, no sé cómo describirlo. Los hemos visto en lo que son estos sectores, y en el Dorado hemos visto otro tipo de pulpos del género Musoctopus, y estos todavía no conozco el género. Tendríamos que tal vez preguntarle a la doctora Janet más tarde. Bueno, aquí ya tenemos lo que es la medida de el flujo de calor, y vamos a continuar entonces moviéndonos para llegar al último punto y poder tal vez colectar algunos octocorales y algunos otros organismos bénticos que nos interesen. Ya veremos que hemos estado viendo algunos octocorales interesantes acá, y lo que son los pepinos de mar, que también ha sido una de las colectas que hemos estado realizando. Les comento que se cuentan con todos los permisos para las colectas y los especímenes recolectados no son antojadizos, sino simplemente es porque no existen. Bueno, estos sitios son poco conocidos a nivel mundial, así que las especies, ¿verdad?, sirven no solo para los investigadores que estén acá o en colaboración internacional, pero también a otras personas de otras partes que los solicitan. Ok. Acá vemos una estrella de mar bien grande. Is that a big one, right? It's a sea star there. It's a big one. Yeah, it's very beautiful. Saludos a mi familia, que todos están ahí conectados, conectadas. Ajá. Esa es una estrella de mar muy grande. Es increíble todo lo que es la riqueza marina en estos sectores. A esta profundidad estamos a 2.950 metros de profundidad. Allá vemos una esponja de vidrio que les llaman, de tipo hexactinélidas. Yeah, it's great. Mm-hmm. So we are approaching some of the bottom is becoming like more rocky. So yeah, some of these pillow lavas here. Can you take a frame grab there with the pillow lavas? Wow, so we are approaching, yes. There are a lot of glass sponges here. Yeah, we have to look for shimmering water. So please pay attention. If you see some shimmering water, just let us know, please. Aquí estamos buscando por agua que se vea un poco distorsionada o alguna salida de agua. Así que estén atentos y atentas. Por si ven, nos avisen. Los dejo apreciar este fondo rocoso con lavas tipo almohadilladas. Siempre el mismo tipo de roca, son basaltos. Es a crab. ¿Vieron ahí un cangrejo nadando? Uy, ya iba nadando, se fue.
Lots of good nooks and crannies. So the shimmering water that we're looking for is similar to like the heat waves that you would see off of the hot hood of a car or on hot pavement.
La profundidad que estamos en este momento son 2.939 metros y estamos en búsqueda de una donde haya salida de agua un poco más caliente, y generalmente este tipo de agua, cuando es la salida, se ven las rocas bastante limpias, digamos, no estaría llena de sedimento porque al salir el agua pues como que limpia la superficie. Entonces esos son sitios bastante interesantes para poder digamos, medir el flujo de temperatura. Esto le llamamos lavas en almohada o en almohadilla, y generalmente se forma en la salida de las dorsales oceánicas, la salida de agua, el contacto con el agua, se forman ese como tipo almohadas o que se ven bastante redondeadas, y se les forma una cubierta más oscura que tiene vidrio volcánico. Entonces se van acumulando así un montón de almohadas, como ven ahí, esta imagen bastante bonita de lavas en almohadilla.
So you can see it got a lot steeper here. So I'm not going to take over the whole screen with it, but I will just pop up the map here briefly in the corner. And I think the ROV is slightly out of date on this. I can update that, but we're headed up that slope on the left side there. So we are past waypoint 9 on the way to waypoint 10. We're working our way up that slope.
Oh yeah. This yellow sponge. If there's another camera there, or we are approaching, because I don't see it. Oh, okay, yes. Okay. Ok, aquí tenemos una hermosa esponja de estas amarillas que hemos podido apreciar en algunos otros sitios. Hemos visto son bastante pocas las que se encuentran. So look at the yellow sponge here. That's so beautiful. Yeah, so we have here like very nice pillow flows. Yeah, pillow lavas. Todos estos lo que estamos viendo son esponjas de vidrio muy comunes en este sector, como más cuando tenemos mucho más lavas, entonces se adhieren a las rocas, y las hemos podido observar bastante en esta expedición. Seguimos viendo estos flujos basálticos muy, muy bonitos, muy interesantes. Estas manchas como más oscuras que se ven, esos organismos como laminares, podrían ser un tipo de briozoarios, parece que sí, o algunos, o a veces se pueden observar también algunas esponjas muertas, como estas que se ven acá, con este patrón reticulado. What's that? Ah, yes.
Looking weird though.
Mm.
Yeah.
How do you call it?
Blanket.
How do you call it?
Acorn worm? What is that?
Okay.
Yeah, that's acorn worm. It's a bizarre.
Acorn worm. Ya les busco el nombre en español de este organismo. En inglés le dicen acorn worm.
Thank you.
Es un tipo de gusano. No encuentro el nombre. Tal vez si los biólogos me ayudan. Algunos octocorales acá se observan, como les mencionaba, aquí en el fondo oceánico tenemos hermosos colores variados, como han visto, organismos rosados, verdes, amarillos. Los colores siguen siendo importantes acá en esta oscuridad. Esta luz que vemos es artificial, es la luz del robot, pero que hay una completa oscuridad. Esto le llaman bellota. Exacto, esos este organismo morado que vimos antes como un gusano grande, le llamamos un gusano bellota o enteropneusto. Es unos grandes pillows acá. Diversos tipos de esponja, por lo menos 3, 4 especies de esponjas acá. Lo que tenemos que cuidar y seguir observando si vemos algún tipo de agua distorsionada para poder así medir flujo de calor. Yeah. Okay. Yeah, it's perfect. Bueno, los dejo con nuestro siguiente colega acá a bordo, el doctor Gustavo Ramírez, microbiólogo de la Universidad de California. Así que los dejo con Gustavo y muchísimas gracias por estar conectados. Nos vemos mañana. Saludo. Let's get him this. So we are going to waypoint 10.
Yeah.
And
So you got a heat flow measurement, right?
yes, we already
Oh,
got some
wonderful.
measurement. So we passed it from the sediment to the pillar lava here. So maybe we can get some heat flow measurement, I don't know. And then
How
we
warm
can
was
start.
the heat?
How long?
How warm? What temperature?
Well, I don't know the temperature. It's warmer? It's getting warmer.
It's getting warmer,
Sure,
okay.
yeah. So we have like an outflow out. And we can start collecting some samples.
Yeah, oh, Kobe, thanks, thanks. Thanks, Tom.
Yeah.
Good afternoon to those of you that are just joining us. My name is Gustavo Ramirez. I'm an assistant professor in microbial ecology and computational biology at the California State University in Los Angeles. I have the honor of being part of a cruise or an expedition. We are calling ourselves the Octopus Odyssey Expedition on board the amazing research vessel Falcor 2, managed, of course, by the Schmidt Ocean Institute. So we find ourselves about 150 kilometers or so off the Costa Rican port of Punta Arenas, and we are a couple of days into the exploration of interesting seafloor features known as seamounts or outcrops. There's a couple technical distinctions to be made there, but previous work had been done in the area in one of these seamounts or outcrops named El Dorado. So a couple colleagues of ours, maybe 9 or 10 years ago, were here sampling shimmering fluids from an incredible formation. Again, it's a relatively small formation called an outcrop at a site called El Dorado. And what they discovered there was quite interesting. There was plenty of evidence of relatively warm fluids emanating from the crust and exiting into the colder water, again, around El Dorado Seamount. These warm fluids have a couple of very interesting and visually striking ecological repercussions. Primarily, the fluids appear to attract octopus females. The fluids of the rate of fluid flow there basically cleans up the otherwise sedimented rocky surface. So when there's fluids flowing, the rocks tend to stay free of sediment, and that appears to be a trigger or something that brooding octopus females look for in order to commit a site to anchoring their clutch of eggs. So that behavior, again, was first described close to a decade ago in previous work that was done in one of these seafloor features called outcrops and seamounts at a specifically place called El Dorado. So with this expedition in 2023, we were charged with revisiting, coming back to El Dorado, of seeing if, number one, the octopus colonies were still there. And we were able to confirm that in our first dive. Another big enigma from the initial exploration here was whether or not the octopus eggs that were observed 10 years ago or so were viable at all. The ROV that was exploring here 10 years ago was equipped with great cameras and had very good close-ups of the eggs, but it was hard to tell if any of those eggs were viable. No biological samples were taken 10 years ago. This time around, again, on our very first dive, we confirmed, number one, that the females were still brooding their eggs at the site of fluid discharge in Dorado, and also confirmed that the eggs were indeed viable. We zoomed in on a couple of them and were pretty much able to see eyes staring back at us. Within minutes of seeing viable eggs, we were also able to see baby octopuses flying out of the nursery by the dozens. So that was a major observation or a major discovery that was made on the very first dive of this expedition. And that is, again, we confirmed the viability and the fact that the octopus communities at El Dorado Outcrop are basically thriving. Something that was previously hypothesized, but actually not known. That's cool. The star is eating the sponge. What we're seeing here is most likely a star feeding on a sponge. Does it have its stuff on the outside?
It does look like it does. Yeah. On the left there. Okay, cool. Thanks.
Thanks for the shout out, Cat D. Right, it is unfortunate that we continue to have weather days in Los Angeles, but I'm looking at the forecast here. That should dissipate soon. Our summer will indeed begin. Sick and tired.
I think we can say we've been to waypoint 10.
Yeah. Okay.
Go ahead.
You're good with that?
Yeah.
Yep.
You're of course here. Thank you.
All right, so just pretty much due south then?
Yeah.
Yep.
South southeast.
Whatever, we just want to go king of the mountain, right?
You know it.
Queen of the mountain.
That's you.
We got a sword to stick on next.
La Jefa.
I am joined in the control room by probably a very amazing collection of minds here. Our chief scientist, Beth Orcutt from the Bigelow Laboratory for Ocean Sciences is here in the dive. I'm also joined by my colleague, Myla Guillon from the Helmholtz Center for Potsdam. She's a research assistant there. And Professor Rachel Lauer, physicist from the University of Calgary, and her doctoral student, Rob Perrin.
It's like there must be a shorter name. And you've got a chance.
We have officially reached the 10th waypoint of the 10 waypoints that were planned for this exploratory dive. again, we are at a feature that is basically a short seamount, technically called an outcrop, that's just east of a larger feature that's been previously identified as Caballito.
Get as much cover as possible.
The primary goals for this dive were to collect temperature measurements all along the seafloor here to inform physical models of how water flows in and out of these seamount features on this particular place of the seafloor. So temperature measurements, again, tell us how water flows. And again, this very simple physical observation, it's really at the core of all the other interesting science that we hope to accomplish here. Water coming in one seamount and exiting another is the most important driver for the emergent biological, geological, and geochemical properties that that the science party is interested in studying. So if we have no fluid flow, there tends to be not as diverse of an ecological community, although that's a hypothesis that we're here to study, of course. What we do know is that wherever fluid flows, there's very charismatic, large life forms and complex ecology to follow. Again, we're past our last waypoint, scheduled waypoint of this dive, and we're simply gliding slightly above the seafloor here. You see a rubble-like environment here.
It does go fairly high. I really want to
That's
check that
just
already.
loaded with sponges and other invertebrates. Octocorals, those really long structures. They can be up to 1.5 meters tall, if not more. They are beautiful octocorals. And
Mostly like a rubble field, so I'm hoping up above it's a little bit more intact.
we've come across similar habitats in previous dives at a seamount called Tangle Set that we explored for 2 days. With our second dive ending yesterday. I made this commentary before, but I'll make it again. I'm just flying around this barren, desolate, seemingly desolate landscape full of sponges and corals. Really does remind me of taking a drive across a Sonoran desert and coming across a saguaro forest. It's really quite striking, really similar, at least in the way that it makes me feel. Yeah. Going to the top, right? Connor's, Connor's asking about our navigation plans here, and we are indeed trying to get to the top of the part of the seamount that we find ourselves in.
Sorry to clarify, we're not going to the main summit, we're going
Right.
to a summit.
The local summit, the local peak.
Yep.
Yeah, so we're at a bit of a saddle now, Connor, and I guess we're based on the closest high point of elevation that we're going to get to is where, or that we can get to here with our remaining dive time is where we're going now. But the actual summit of the entire edifice, we'll have to come back and visit perhaps tomorrow. again, we find ourselves about 1/10 of a degree of longitude east of a larger seafloor feature that's putatively named Caballito, or Little Horse. We speculated yesterday, looking at the low resolution bathymetric map, on why this structure may have been called Caballito. It appears to have a northern depression that actually looks like the head of a horse, or perhaps a knight on a chess set. So again, complete speculation, but again, that's a larger feature just west of us, about 1/10 of a degree in longitude west of us, that we will visit either tomorrow or the day after that. So we are on an unnamed feature, so just east of Caballito. This feature and Caballito, again, are known from bathymetry data, and some, I guess, the temperature here has been hypothesized based on very relatively old temperature measurements that were made from a ship literally lowering a thermometer to the seafloor from the boat, and then taking basic measurements of the sediment temperature in this area. But human eyes have never explored either of these two seafloor features before. Our dive is the first one to reveal to humankind what these seafloor oases actually look like. We can see the terrain getting steeper as we continue our ascent, and away from a saddle point here and into a local high point of elevation. Again, we are aware that the feature has a summit that's further to the north of us, and we will not summit it today. The plan is to perhaps come back and summit this tomorrow, but that is still TBD. We will have our scientific meeting immediately following this dive, this live broadcast, and see if there's a scientific merit in summiting this feature tomorrow. Connor is asking, when were they named? So there's been some discussion about this, Connor, amongst the science party. So it's just old bathymetry and temperature data literature that I believe comes out of the 90s. So I'll get back to you and have a closer look on that, Connor, or about that, Connor. Our Costa Rican colleagues were definitely interested in attempting to name some of these sites. So we're beginning to look into the formal process of doing that. It's not as simple as calling it what you want and then publishing it with that name. Those names remain putative. There is a governing body authority that we need to write a short proposal to, and then and that's responsible for validating the official names of some of these seamounts. So we're definitely looking into that. Got another one of these long fishes hanging out. You know the name, Klaus? Fish. Oh, it's gone. All right. We're zooming right past here, and that's okay. That's exactly the plan. We are not in biology mode. We are in survey mode. So we are, again, in the few minutes that we have left of this dive, half an hour or so, we are just gassing the ROV up to the local summit here, away from a saddle in the topography. Whoa, another large sponge there in the back. Yeah.
That's enormous.
Right, the geology hasn't changed hasn't changed drastically in the last couple minutes of the dive here. However, something appears to happen biologically. There's just more sponges on the seafloor the further up we appear to go. Right before us is a behemoth Can we get lasers? Is that about a meter? Very briefly, I know that that's not our priority, but just out of curiosity. We're getting frame grabbers. Crazy cool. Okay, I'm having a hard time seeing them. Oh, I see it. Okay, yeah. So for those of you watching at home, you'll see two small green dots rising up along that giant sponge there, and those 10 dots are 10 centimeters apart. So just eyeballing what I'm seeing here, I can confidently say that this sponge is close to a meter in height. Yeah. And it's, of course, adorned with this coral at the very top, looking like a head ornament or a crown. Wow. We can carry on. Sure. This is a priority. Yeah, it's just, I'm just looking for fodder for the narration, but it's not that hard to find. It's everywhere. This is super cool. White thing in the back. Yeah. Thanks for the feedback there to those of you on the chat. So the sponge did not have a coral. Cat D says it was wearing a fascinator. That was indeed working. Yes. Again, just an explosion of biological activity here. Our octocoral expert and also our chief scientist present here in the control board with me have instructed the ROV pilots to focus on this structure that's potentially a coral.
We saw one earlier that was kind of broken. It's like the bubbles down the corner. There's a really nice specimen right there. Yeah. This side's
Go ahead and take the time now to do a brief summary of the action that we've been experiencing over the last 20 minutes in the Spanish language. Buenas tardes a todo el mundo de habla hispana desde las aguas a 150 kilómetros de distancia del puerto de Punta Arenas, Costa Rica. Bueno, me llamo Gustavo Antonio Ramírez Ábalos, soy profesor asistente en la Universidad de California, Cal State University en Los Ángeles. Y tengo la dicha de encontrarme a bordo del buque de investigación Falcor 2, operado por el Schmidt Ocean Institute. Y bueno, venimos equipados con un submarino remoto llamado SuBastian. SuBastian es una maravilla de la tecnología moderna que nos deja bajar cámaras de ultra resolución al lecho marino e investigar con nuestros propios ojos remotamente, obviamente, todos los misterios que existen ahí.
We want to make sure we get a land shot before we take it.
Estamos estamos trabajando aquí como parte de una expedición oceanográfica llamada Octopus Odyssey. Y la misión de esta expedición es básicamente entender, entender las razones físicas, geoquímicas y biológicas por las cuales las hembras de pulpos del mar profundo prefieren resguardar y plantar a sus huevecillos en piedras justo arriba de lugares donde aguas hidrotérmicas salen del lecho marino. Ese fenómeno fue descrito por primera vez hace aproximadamente 10 años y bueno, nosotros venimos aquí a confirmar que esa actividad siguió ocurriendo en una de estas montañas submarinas donde se documentó hace 10 años. Durante nuestro primer buceo pudimos confirmar que las hembras seguían ahí con sus huevecillos y más que eso también que los huevecillos eran viables y que esa pequeña guardería de pulpos o jardín de pulpos contiene pulpos que están felices con comunidades que están activamente reproduciéndose, ya que vimos docenas de pequeños pulpitos o pulpos bebés que acaban de nacer. Después de haber confirmado eso en un lugar que se llama El Dorado, una pequeña montaña submarina que se llama El Dorado, nos hemos dedicado a seguir explorando la vicinidad del Dorado, donde se encuentran muchas otras montañas donde potencialmente también este fenómeno hidrotérmico puede atraer pulpos para hacer sus jardines y para depositar sus huevecillos. Esta es la cuarta montaña que exploramos en el área después de haber explorado El Dorado. Y bueno, es importante recalcar que aparte de la montaña El Dorado, las otras 4 las cuales hemos visitado nunca jamás habían sido vistos por ojos humanos. O sea que esto es 100% exploración. Obviamente existe una hipótesis que nos trae al área y que inspira las metas científicas de la expedición, pero verdaderamente con cada buceo nuevo que hacemos estamos explorando partes del lecho marino que nunca se habían visto por los humanos. O sea que es extremadamente exciting poder visitar estos lugares y ser los primeros ojos en toda la historia de la humanidad que ve las maravillas que la madre naturaleza esconde aquí. Bueno, cualquier hispanoparlante puede hacer preguntas en el chat. Nos encontramos me encuentro con varios colegas costarricenses, especialistas en biología marina, oceanografía, en microbiología, sedimentología. O sea que cualquier pregunta que exista, pueden preguntarnos. La última cosa que recalcaré en español es que la mitad de la expedición está compuesta de científicos costarricenses. Estamos en el Pacífico costarricense, o sea que es importantísimo de incorporar o de hacer estas exploraciones con equipos diversos y multinacionales de Latinoamérica. Más del 90% del territorio nacional de Costa Rica es en sí el lecho marino profundo, o sea que es una misión importantísima, especialmente para nuestros colegas ticos, el investigar lo que ocurre en lo que es físicamente la gran mayoría de su territorio nacional, el lecho marino. Okay, I'll go ahead and carry on with the English narration. We are about to collect appears to be an octocoral sample. And there's some, octocoral sample confirmed by our Odelis Cabridi, our University of Costa Rica colleague here on the ship with us in the control room, leading the logistics behind the best possible method of collection of this specimen with the 2 ROV pilots. So the ROV is equipped with two arms. One of the arms has got a bit of more brute strength. It's a triple prong arm. The arm that you're seeing on your screen now, it's I guess it can be used to handle more delicate samples with a bit more finesse. Oh wow, got a what was that, an eel or fish? Photo bombing our collections here. Yeah, ooh, look at that. Se escondieron todos. Sí. Collaride? It's the first time. You go ahead and say here. Couple of some information about this specific collection from our local our onboard expert, Odalis Caballito, professor at the University of Costa Rica.
This is the first time in the expedition that we are going to collect a specimen from this family, probably Corallidae, and we have seen two others with different color, but this is the first one that we are going to collect. Vamos a recolectar por primera vez un octocoral de esta familia, probablemente Corallidae. Hemos visto otros morfotipos, pero esos no los hemos recolectado todavía.
Again, that was a commentary by our octocoral expert, Professor Odalis Caballito from the University of Costa Rica. Is that a bubblegum? Is that a bubblegum? No.
Bubblegums belong to the same family.
Same family.
Yeah,
Same family.
bubblegum. Yes, it is.
Yeah. Brett's asking if I'm into cave diving. How you doing, Brett? Believe it or not, I am not a scuba person. I guess the few times that I attempted to dive, I guess snorkel beyond 5 meters, I had a lot of problems with my ears. But doesn't mean that I won't try it again. I guess I will remotely dive anywhere in the world if I can get an ROV team to come and join me.
You know our number?
Yep, I indeed know their numbers now. So yeah, I haven't personally dove. I should mention that I've had the privilege, however, of having one Alvin dive back in the summer of 2014. So I was one of 3 people that descended on the remotely operated vehicle Alvin to a depth of 2,693 meters beneath the waves at a very special site, very close to my heart, called the Juan de Fuca Ridge. So that's most of, or that site is basically where I did most of my doctoral work, or experiments from that site were most of my doctoral work under the leadership of the late Katrina Edwards, and actually after her passing, under the leadership of our current co-chief scientist on this expedition, Dr. Beth Orcutt from Bigelow Laboratories in Maine. So that was pretty cool. That dive was literally changed the way I see my own life. It's surreal. Going down to the seafloor on your own, it's I would say almost pseudo spiritual experience. You know, nothing will feel the same afterwards. Yeah, I wish that honestly that we could get congressmen and the general public to visit the deep sea floor on in Alvin trips, so that they could understand the immense beauty, but also very, very vulnerable fragility of this gigantic ecosystem that's literally 99% of the habitable space of our planet. So, so anyway, thanks for asking, Brett.
Bend it in there, like try to push down and bend it. Yeah, so maybe to just trim off a little bit. Yeah, I can just push down over here at the very tip. Hope for the best.
Is that back corner in the top forward part?
Under the watchful eye of our octocoral expert, Professor Odalisca Bridi from the University of Costa Rica, our masterful ROV magicians here, I guess J-Rod specifically, collected, just finished a fantastic collection here of a beautiful octocoral specimen. And of course, the goal of this collection is to minimize the structural damage that we do to the specimens when we're putting them in our biological collection boxes. And, again, skillfully, extremely skillfully done by our ROV pilot group, SuBastian ROV pilot of the Schmidt Ocean Institute. That specimen is going to be a paper already, guaranteed? Yeah, first of its kind?
I don't know if that one is new, but to analyze it. Actually, I have one that is sweet. I remember the one that we collected. This is different from the other that I'm describing to focus.
Got it.
So for sure it's not on this one.
Okay. Yeah. I don't know. That was it. I'm not seeing what that was. I don't know. No sé si qué quiere hacer Beth específicamente. Yeah. You have to do a couple, yeah. Okay. There's this one right here. There's one of those real brittle ones.
What is this? That we are kind of yes.
And then we hit waypoint 10. So we just saw that and got it right now. Hey, you want to do a quick summary in Portuguese?
No.
No? No? Okay. Yeah, tomorrow. Well, we're welcoming you to the same thing.
Yeah, okay.
Jump in. Come on. So you should want to get one more quick sample and then come up?
Yeah, do you think it's possible on the way up to try test firing one of the squeezer samples in some of the box? We don't need it on the ground, just anywhere. I just want to see if it fires.
Absolutely.
Because we've been having trouble with that. Just thought about it now.
Seems like there might be a chance for one more quick collection at the seafloor here before we blast off and start heading back up about 1,800 or 2,000 meters, or 2,843 meters back up to the surface world.
Yes, it's the same one.
Same one?
Yes, it's the same. You can take a
Our ascents are also action-packed. We were able to see some a lot of action in yesterday's ascent. We expect the same today. Perhaps the two highlights of the hour-and-a-half ascent from yesterday when we left Tengo Set Seamount were a squid that delighted us by skirmishing away with an inkjet right in front of our camera, and also a gigantic, perhaps 5 or 10 meters across, siphonophore. These are colonial animals that basically make giant chains of clones of themselves that can be tens of meters in length sometimes. So we got a chance to see that too yesterday. So if you're a pelagic person, not as much of a benthic person, feel free to stick around and see what what mysteries the water column holds in store for you. What excitement! the ROV release valves and then come up, or
No,
it's always rising?
Yeah, so we usually try to put it slightly positively buoyant for a couple of reasons. But
Drop weights and
no,
just goes?
no. Yeah, it's all propulsion.
Just
fly it.
Okay.
Yeah, we're just gonna test fire this one of the squeezer samples because we've been having trouble with that. I want to see. We adjusted the bottom to see if that would work.
No sample?
No sample. We're good. So we
Okay.
can stop that. Yeah.
Grab one of those real quick here.
Yeah, we'll do. You start. Okay.
Again, we're coming on to our last sampling attempt for this dive before blasting off from this unnamed site east of another site that's also unexplored called El Caballito. We're 150 kilometers more or less offshore the Costa Rican port of Punta Arenas. We're off. All right. And we are off.
That's off bottom.
This concludes the first remote exploration of this previously unexplored, unnamed seafloor feature, small mountain of rubble, located about 1/10 of a degree east of a larger seafloor feature that's also unexplored, but that's been named in the past, at least in the literature, called El Caballito.
I can do it if you need. Oh, just while you're setting up, you can.
We shall return. We leave in peace for all mankind. And as I was mentioning earlier, our ascent can be action-packed also. We were able to see a squid yesterday, tons of invertebrates, bioluminescent invertebrates, and also perhaps the highlight of our ascent yesterday was a gigantic 5 or maybe 10 meters across, we're not sure yet, siphonophore, so a clonal organism that can be tens of meters in length. The first order of business during the ascent here, as suggested by our chief scientist, Beth Orchid, is the test firing of something called a squeezer sampler. So these squeezers that you see right in front of you now on your screen are custom-built large syringes that can be triggered by an ROV manipulating arm, as you'll see here. Those white plastic parts have a triggering mechanism that allow the plunger that you see at the bottom of the screen there to move up, and by doing so create a vacuum space that brings up water from wherever this is triggered. So again, we've had slight bad luck with a couple of these. They were not triggering at the sites where they were intended to be triggered. I think yesterday and perhaps the day before. And again, the goal here is to see what's going on with these. You guys squeezed it? No go? No go? Okay. So this was a no-go. The arm there, the manipulator arm, the left manipulating arm was supposed to give that or it did give that a squeeze. And the plunger there did not retrieve. So there was no sample collected. So that was syringe number 3 here. So I guess we'll try a couple of more as we are on our way up to the surface.
And we've got about 45, well, about 40 minutes or so now before the stream would need to get reset. So even once we wrap up here, I'll leave it on until we're closer there. We won't make it all the way up. I'm not even sure we'll make it half the way up. But I know we've been seeing some fun stuff on our ascents, so I'll leave that on.
What's that? I think that milk tray has just been, you know, not treated so sweetly. Can you get an angle on the divider? So you can have more room on the inside. I have to rotate it. There you go.
And we just saw the masterful rehoistering of a squeezer number 3 there by our ROV pilot team. Not an easy task. It's a pretty snug spacer where that squeezer is supposed to be replaced. And I mean, it's 6 screens, 6 two-dimensional screens that the pilot has to watch at all time to give him a 3-dimensional perspective. So beautiful.
Yeah, we pull that up so we can see the number on the side.
So what we witnessed there was a successful test of these squeezer samples, squeezer number 9, that upon triggering by the left robotic arm there, squeezing the plastic plates, I see there, the white plastic plates, that released a trigger that brought that plunger up, and therefore in the space that was left behind by the plunger, there is now water that will sample a depth that we will then be able to examine here back on the ship. So again, we're going for a 3rd squeezer test here. Bueno, ahora sigo ahora con un poco de narración en español. Hemos despegado de la cima de una montaña sin nombre que se encuentra en el lecho marino en las aguas costarricenses, justo al lado de otra montaña que aparentemente tiene un nombre, aunque es un nombre extraoficial, el Caballito. Y bueno, lo que estamos aprovechando el tiempo de ascenso de aproximadamente 1 hora y media, desde una profundidad de más de 2,000 metros y medio debajo de las olas. Estamos aprovechando este tiempo para hacer pruebas de unas jeringas especializadas que se llaman squeezers. Estos squeezers, son básicamente jeringas que tienen un sistema de detonación plástico, que es donde está la mano izquierda del robot, que pueden ver en la pantalla, sosteniendo el squeezer. Y al aplicar presión a este sistema plástico, se supone que la jeringa se retracta, y al retractarse absorbe agua del orificio del orificio que se encuentra bajo la jeringa. Bueno, esta fue la 3ª prueba. Son 4 aparatos que tenemos a bordo y solamente uno ha funcionado. O sea que y no es tan fácil como simplemente hablarle a una compañía donde compramos eso, nada de eso. Esto es equipo para trabajo súper especializado que se tuvo que diseñar por los propios científicos que están a bordo conmigo aquí en el Research Vessel Falcor 2. Y bueno, esto es parte de su control de calidad. Estamos viendo si la configuración de las jeringas aquí, o squeezers, es apropiada para que sean detonadas apropiadamente la mayoría del tiempo estas profundidades. Bueno, vamos por la cuarta ahora. Number 1. Squeezer number 1 will be our fourth test of this very sophisticated yet simple syringe system. This robotic arm again is going for the white plastic lining there. That's supposed to once pressure is applied to the white plastic lining there, the triggers release mechanism that allows that plunger to be retracted and for a sample to theoretically enter the collection chamber. And it looks like this one failed to fire again. Okay, this is a third misfiring, or out of 4. Again, this for all tests of this specialized equipment. Our scientists on board are basically designed and build these specialized syringes for the special experiments that they want to perform at the seafloor. So it's not as simple as calling up manufacturer and telling them to fix this. But it's scientists that have to go back to the drawing board and figure out how to best get most of these squeezer samplers to work on the next dive. So that's the plan here. J-Rock, this just light diffraction, right? It looks like a fake seafloor.
Yeah, it's just, the way, all the lighting on the stuff is designed for that camera.
Yeah.
So this is up near all the lights. Yeah. So you're just getting all that side light coming into the dome.
Got it. Okay, yeah. Just a little bit confused by that because I saw the depth meter. We're definitely. All right, with that last bit of action, I will go ahead and sign off as the narrator for this dive. Again, my name is Gustavo Ramirez. I'm an assistant professor at Cal State University in Los Angeles in microbial ecology and computational biology. And I am joined on this expedition, the Octopus Odyssey Schmidt Ocean Institute expedition, by a number of international researchers. At least half of the members are from Costa Rica, the host country for this work. And we have about an hour and a half ascent back up to the surface world of our ROV SuBastian here. Again, for those of you that are going to stick around and look at the ascent, there's some action guaranteed. Definitely invertebrates, plenty of bioluminescence we expect, just a couple hundred meters above here. And yesterday we were extra lucky. We were able to see really cool sightings. But for the meantime, I will step away from the dive lead and commentator chair and sign off from all of us on board the research vessel Falcor 2. Have a good rest of your afternoon and see you tomorrow. Our dives began at 7 a.m. Local San Jose, Costa Rica time. Take care, everyone.
All right, like you heard, we have quite a long ascent ahead of us. And as I mentioned previously, we've only got about 30 minutes left on this stream. So we'll see what we can see in 30 minutes. But otherwise, we'll see you all tomorrow. So I'm going to leave the stream on. And I'll do one final little sign off in about 30 minutes, no matter how high in the water we've gotten. And we'll see what we see. right. So as we come up on this solstice, nice jelly in the water. J-Rod getting us some good blue water. This will be the last thing. But I do want to thank everyone for joining us and coming along and exploring with us. We will be diving again tomorrow. That's the plan. About the same time. Getting on the bottom and starting the stream at about 7 a.m. Local time for the ship, Costa Rica. Lovely jelly just sitting in the water. Saw this. Couple siphonophores. Little isopod of some sort. Oh, yeah, we saw the chat debating whether or not it was a radiolarian. Subsea chestnut is J-Rod's official name on that one. But we'll be back tomorrow. Thank you all for joining us. Enjoy the rest of your day, your morning or evening, wherever you might be. And thank you for exploring with us.